I'm trying to skip. I tried to have something about it. So I'm just trying to skip. It's difficult to know. It's a very such a cool inspired. Yeah.
Thank you. Siddiqui.
Do you start now? Yeah. Good morning, colleagues and guests. When the African drums beat, they beat to celebrate events and festivals, to bring healing, for spiritual rituals, for sending messages over long distances. Our drums beat today to warmly welcome you, colleagues and guests, to the 19th Annual International Biocuration Conference, the first of its kind to be held in Africa. My name is Vicky Nembaware. I'm from the University of Cape Town. It is an honor to co-host such a diverse group of professionals dedicated to the field of biocuration. Over the next 3 days, we will explore cutting-edge developments in areas pertinent to biocuration, such as data integration, annotation standards, knowledge representation, and the application of computational approaches to support ethical, scalable, and reproducible science. We hope the conference provides a stimulating environment for meaningful exchange, new partnerships, and innovative ideas. Please project the slides. Welcome, Mauiuya, once again. Siakwamukele. Our sincere thanks go to the partners and sponsors. This meeting would not have been possible without their support.
Okay, good morning, everyone. My name is Lyndon Zass from the University of Cape Town. You've probably received many an email from me over the last few months. I do apologize and thank you for your patience. Regardless, you might receive a couple of more from me over the next few days. Before we begin the program today, I'd like to thank some key individuals for helping us curate the conference program. So firstly, we've got the scientific committee who've joined us over the last several months and helped us review abstracts, curate the conference program, and also plan the event logistics. Can we have a round of applause for them, please? Secondly, we've got a very amazing local organizing team. So I'd like to give them a very, very big thank you. They've been working behind the scenes all week and over the last few months, but they've also been working a bit in front of the scenes as well. So I'd like to thank them as well for all their hard work over the past few months, and I'd like to ask everyone to extend an applause to them as well. Okay, to get into the conference program a bit, over the past 2 days, we've held 11 biocuration-related workshops, and over the next 3 days, we will have 11 key sessions as well. Within these key sessions, we've got 40 oral presentations, 2 panel discussions. We will have over 25 posters, both online and in person. And then next door, we've also got 4 exhibition booths. So I do encourage everyone during tea breaks, please visit the booths, visit the posters, and engage with our presenters. Regarding our delegation, we've got 134 official registrations. We've got 20 countries represented officially. This is based on affiliation, but I'm sure we actually have a lot more represented in the room as well, and you'll get to know them over the week. We had the pleasure of sponsoring 8 travel fellows from across the world to attend this event, and then we've also got 20 high school learners and educators joining us today as part of our high school engagement and outreach. So this is the illustration of our delegation this week. Like I said, this list is not exhaustive, but what I really want to highlight and what I'm proud of is that we've got 9 African countries represented here this week. This conference represents an amazing opportunity to expand the biocuration community within Africa, but also to introduce the Society of Biocuration to African biocurators. So with that, I'd like to thank all the conference delegates for for adding to the conference program. And I'm sure we'll have a very successful, engaging, and productive Biocuration conference this week. Thank you, everyone.
Thank you once again to you, the conference attendees, for unknowingly supporting prizes for the high school learners quiz and debating competitions. We would like at this stage to thank the Metro South Education Department, especially Mr. Gabriel Agalas from the Department of Education, who willingly collaborated and has continued to collaborate with us over the last couple of years to work with high school learners. Thank you. So I'll give an opportunity to one of the winners from the quiz and debating competitions that this conference supported to say a few words.
Good morning all. My name is Sarah Dolly and I'm a grade 9 learner at Darul Arqam Islamic High School. I'd like to say thank you for the opportunity to join you guys today. The MSED STEMAC competition has inspired me to pursue a career in science. It started off as just another subject that I was interested in, but now I think that it's my lifelong passion. Thank you.
And a big thank you to the educators who work so hard to give confidence and skills to speak in public to the high school learners. We would like to encourage you to engage with them throughout the day. They are only here today. And during lunch, there will be mentorship tables, which are Dr. Kutala Munika standing over there, who's raising a hand, is organizing. So if any of you are interested in engaging with them on a more personal level, please reach out to her. Please help me to welcome our Dean of the Faculty of Health Sciences, Professor Lionel Green, who's made time and a great effort after a very long trip. He came straight to the venue to support this event and to address this conference. Professor Lionel.
Wamkelekile apha eUniversity yaseKapa. Welcome to the University of Cape Town, because I suspect that even though we're not on the campus, the spirit of the university is very present here. It's literally a kind of bilocation kind of process. There's something though, before I make any words of welcome, there's something about a young person speaking about a lifelong ambition. And I think that when I hear young people speak in the way that the person who introduced this morning spoke, I'm really struck by how we're caught always as a society of scholars in this idea of who we are as people gathered, the kind of processes we embark on, and then seeing the promise emerge in young people like the person who's just spoken now. So I want to congratulate the learners who engaged in the process and just recognize that there already appears to be an openness among you to engage in processes of science and then commit to a journey that you don't quite know where it will take you at this stage. But I think we look forward to walking that journey with you. But good morning, colleagues, partners, friends. I'm really struck at the fact that this Biocuration conference, which is a growing field of enterprise, has attracted as many people from as many countries. So we do want to express a warm welcome to you from the university. But let me recognize those who have been involved in the local organizing committee, the scientific committee who reviewed the abstracts, and just recognize the work that goes into an event like this. When Vicky spoke about when the African drums sound, there's something about the drumming in Africa that calls people to attention, but in this instance announces the beginning of an important conversation, because that's really what you're about here. So let me also acknowledge those partners who've gathered here: the International Society of Biocuration, the African Bioinformatics Institute, the African Genomics Data Hub, and the Sickle Africa Data Coordinating Center. Because I think collectively you're serving to grow a field which is earlier than many other of the disciplines in the health sciences, but is critical. And the fact that you've come together from the globe in an African context says a lot for your recognition of the place and the role of Africa in the conversation. Because in the echo of those drums that were referred to earlier, they add a sense of how powerful this gathering is this morning. And the work that you're about really recognizes this moment of amplification of the work that we're doing in biocuration. The expertise that is gathered here is significant: curators, developers, researchers, clinicians. But it offers us the opportunity of translating your work, or for us to watch you translate your work to make greater impact. Because this is not only about data, it's about the conditions under which that knowledge can be trusted, it can be shared, and it can be used. And increasingly, that's the notion that we need to be engaging. This conference that I returned from in Canada, the opening session was a conversation about the intersection of AI and the communication and information that communities consume. But the guest speaker was, in fact, a person who'd been an anti-vaxxer during the COVID period, who'd gone through the experience of, dare I say, enlightenment in terms of engaging with the science in a different way, and has now become an advocate for how we communicate with communities about what science means and how we interpret the data that we collect. But I think there is a sense of urgency for the African story. Africa is rich in its knowledge, in its biology, in its disease patterns. And when we say rich in the disease patterns, it's almost a contradiction in terms. Because one of the things that I'm increasingly saying to our faculty is that we've got to recognize here at UCT that we're only as good as we are because our patients are as sick as they are. And it's almost a contradiction in terms that our expertise almost relies on the vulnerability of our people. And so as we collect the data and have conversations around this, the onus is on us to change that story so that the people that we look at and whose data we collect are not only subjects of the story, they become active participants in changing the trajectory as we go forward. I've also been using a quotation from Jomo Kenyatta more recently about this idea that in an African tradition, we can continue to tell the stories of our great heroes. And there are many amongst the African pantheon, amongst our madlozis, as we say in isiZulu. But what Jomo Kenyatta says is that we can either keep telling the story of heroes, or we can become the architects of our own future. And so much of what I see in your program is about constructing that future together, that in many ways, this conference itself is an architect, a dynamic architecture that is predicated on the interaction of people together. Because coming to Africa, you recognize that representation within our scientific endeavor has been incomplete. And when I think about the completion of that representation, it is about social accountability. How do we craft social accountability as a commitment, and that everything we do, both in research and education, responds to the needs of our communities? I was trying earlier this morning just to try and understand what stories are being told by Africans about biocuration. And I picked up this idea of priorities or recommendations that Adeyemo Eleyodi, in an article from Nigeria at the beginning of last year, speaks about these 5 priorities or 5 recommendations for biocuration in Africa. And I thought it speaks a little bit to trying to understand your discipline in the depth that you have engaged in. And I see some of those things already on your program, this idea of the integration of indigenous knowledge systems, making sure that when we collect the data and store data and manage the complexity of the data, we recognize the environments from which it comes and the impact on those communities. The investment architect, infrastructure and technology, I think that's an ongoing conversation in our context. That there's a critical capacity building and training imperative, which I think is already being played out in your interaction with schools and with the younger generation. Development of standardized tools and protocols, and then this idea of creation of regional biocuration hubs. I think that it's very clear that that remains a priority for all of us. And so what is encouraging for me coming to this conference at this time is that we increasingly are recognizing the need to amplify the story within the context that we exist. And across the continent, we're seeing that through partnerships with global colleagues, there is important progress. That I think this meeting represents some of that progress, which forms the foundation for the future of health sciences. And it's worth pausing on something else that is quite significant about this meeting. This idea that you've chosen to bring established experts into the company of novice scholars. And if we invest in that young generation, we've no doubt going to build something significant. And so part of that idea that we increase the representation, both in the places we gather for these meetings, but also in the people we are nurturing, it really starts to change the conversation about who enters the field, who is trained, and who has the capacity to lead in the future. I want to argue that your location in Cape Town recognizes the importance of Africa in the story. But I want to say that to the Africans amongst us, that in fact, you're a critical part of generating this conversation and making sure that the conversations you have both within this conference and that you take away are generative in the way we are represented in these conversations. There's a Nigerian proverb that says, until the lion learns how to write, every story will glorify the hunter. And so in many ways, the relationship of communities with science is very similar. We've got to make sure that all the lions learn to write so that we ensure that the story is far more general and is heard in many more different ways. But congratulations to the organizers of this conference. This is a significant moment. To the delegates, I hope that the conference is a meaningful time of growth. To those of you who've joined us from places further afield, it's quite hard to travel, so we're really grateful that you're here and will make a contribution. But really at this stage, Inkosi kakhulu, Kasi kai Kangans, I wish you very well with this conference.
Can we have another round of applause for the Dean, please? Thank you very much for taking the time to spend with us this morning as well, and for welcoming the delegates to this amazing opportunity. Okay, so next I'll introduce one of the executive committee members of the International Society of Biocuration, Dr. Victoria Nunes.
Hi. Hi, everybody. Good morning. I'm Victoria. And in the name of the ISB, we would like to welcome you all to this 19th Annual International Conference, Biocuration Conference. Okay. For those of you that are new to our field and might not be super aware of who we are or what we do, we, the ISV, are now, for a couple of years now, officially a Swiss non-profit organization for biocurators, developers, and researchers with an interest on biocuration. We try and we do our best to promote the field of biocuration and provide a forum for information exchange, for networking, to build a community with each other and have a space to contribute with each other and help out everyone that's part of the field. So we have been supporting local and international meetings in several countries, and also recognize both early career and senior curator achievements, and provide several grants opportunities to our members. This is the 19th, so we have supported 18 annual international biocuration conferences across actually four continents, because as Vicky was saying, this is the first time that we are holding the biocuration conference in the African continent. I'm sorry. So we try to move around to be accessible to everybody all around the world. So we try to rotate the conferences between the Americas, Europe, Africa, Asia, and Oceania. On 2024, we have our conference in India, and last year we have it on the United States. And here we are today in Cape Town. If you want to know more, you are going to be seeing things regarding the ISB all over the conference, of course. We have some speakers, some presentations lined up, but we also have a poster that it's hopefully in the next room, and if it's not there yet, it's going to be there shortly. You can come and talk to us, but of course you can also reach out to us in our variety of social media. You have our handle there for Blue Sky and also for Mastodon. We are in LinkedIn, of course, and for day-to-day communication, you can join us in Slack. And we are very interested in talking to you, figure out if you have suggestions, if you have any problems, or just if you want to know how to get involved, become a member, or become a part of the executive committee. We are here and we will love to talk to you. This time around, you have me and Peter Jutz in person as the ISB representatives, but there's 11 of us, and many of our colleagues from the executive committee are present online. So as I was saying before, we have some moments for the ISB on the lineup. So in a brief moment, you will be hearing about the exceptional contributions to biocuration, the Lifetime Achievement Award that we granted to Professor Ruth Lovering, which is in like 15 minutes or so. And then we're going to be showcasing what we've been doing regarding the annual conference travel grants. You will have an ISB update and also the details of the 2027 annual International Biocuration Conference are going to be disclosed in the closing session. So basically, if you were wondering why it's important to become a member, I think this also highlights this. We and the ISB, we try to give back to the community with grants for people to travel and to attend to these kinds of meetings, and also to highlight the contributions to our fields. So also on that note, and on the fact that this is, again, our first time in the African continent, I think last year, we have started our EDAA office, so to promote equality, diversion, inclusion, and accessibility across the society and the broader curation community. We have our officer, which is Luana Licata, another of our executive committee members. And we are happy to say that this is an hybrid conference, so we have many attendees that are participating online because they couldn't maybe attend in person. So this is another way that we want to reach out to everybody. Okay. And with that, well, I welcome you all again, and we will have hopefully a very good and productive meeting. Thank you. I'm going to leave you now with a video that one of our colleagues from the executive committee prepared as a tribute for Amos Bayrock. This is a video from Frederic Lisacek, who I think it's online. Thank you.
Welcome to Biocuration 26. We thought that we would pay tribute to Amos Bayrock, who left us at the end of 2025, for the very important traits he left on our path of biocuration. And I'm going to not give you a full-blown biography of who he was and how fantastic, whatever. I'd like to keep it simple. You can find a lot of information on the Swiss Institute of Bioinformatics pages, on Wikipedia, on articles on the history of SwissProt, or you can ask an AI conversational agent to find out what he did. What I would like to talk about is what kind of biocurator he was, what did he do in that particular context. And for those who don't know, I'll briefly say that he created and really fed in the most essential way of speaking databases like, of course, SwissProt that became UniProt, ProCite for motifs, Enzyme for enzyme, NextProt for human protein, Silosaurus for cell lines, etc. And he, to do that, co-founded the Swiss Institute of Bioinformatics in 1988, so as to have the infrastructure to support his initiatives. And he also was behind the creation of the International Society for Biocuration, where at the beginning, most people who were active there were part of his environment. So you can have details. There's Alex Bateman published this paper in Bioinformatics Advances 2 months ago, and he will be present at the conference. So ask him some questions about Amos and have more details. For the time being, I want to talk about Amos as he was. Amos was passionate, and you can see that in a video, which is the last video presentation he gave in 2025, July 2025, because he received the award for accomplishment by a senior scientist, and that is an award of the International Society for Computational Biology. And I will only borrow his words to give you his viewpoint on biocuration. Amos had a great sense of humor, so all his slides, there's always a joke. It's never very formally presented. And so in one of his slides, he thought it was important to show what a good biocurator is. So of course, he expects a good biocurator to have a broad knowledge of the field he's biocurating, or curating, I should say. He also was himself absolutely enthusiastic about something new coming up, a new technology, a new discovery, a paradigm shift, anything that would shed some new light on the field you are working on. So he was working on. It's important to want to keep on learning and be happy with new learning. Not to be discouraged by the fact there's a ton of work and that a ton of work will remain a ton of work. And so this is what it is. So we have to do what we can do. And the ton will never reduce, but still a little bit, a few kilos here or there. He was praising a synthetic mind, of course, which is important, and an ability to spot errors, which is really important. When you read a paper, you have to see where if there are errors and where they are. So his words, samurai drive towards perfection. So he was himself really wanting to be as good as possible. And of course, it's not possible to be perfect, but you can strive to go in that direction. And then his expression, maybe unexpected artistic attention to detail, means probably the obsessive look at details if you want to really understand what you're doing. So that was the first slide. The second and third slide I borrowed from this presentation was the way people think of biocuration. So he first targeted the funders, and the funders were totally unimpressed in the 1980s. In the 1990s, they thought, maybe this is interesting, but they had no idea that it was an ongoing series of tasks. At the turn of the century, of course, text mining was very popular, and so it was thought that it could replace manual curation. In the 2010s, then he had the idea that crowdsourcing would be a solution. So create those communities of people and have them really co-annotate, co-curate data. That worked for in some very specific cases, but generally it wasn't. And then, of course, with the dawn of the rebirth of AI and the deep learning methods in the 2020s, everybody thinks that manual curation can be replaced. But of course, listen to this video and you'll see that he has a very critical point of view on everything. And in this third slide that I borrowed, he's explaining that people have different viewpoints. Of course, at the beginning, students and PIs didn't see exactly the same thing. PIs were very unimpressed, students as well, to be honest, 50 years ago. And then students became enthusiastic and PIs started to think, oh, okay, I can put a junior scientist maybe on a topic like that. And as time went, a ton of work remains a ton of work. So everybody started complaining that we were too slow, or everybody is thinking that now with the availability of many computer means, probably we can automate it all. And again, listen to Amos. That's not his view. It's very sarcastic to put it that way. So just to finish, because I don't want to take that much of your time, I finish with digital Amos. Amos was absolutely passionate about new technologies, whether they were digital or in biology, in biochemistry, or he was interested in astrophysics. He was interested in everything. And you would send him a picture after a dinner saying, oh, we had a good dinner. This is what he would do. Find an app to do something that was different. So that's his sense of humor. That's the way he was. And he was also laughing. We laughed with us. He laughed with us. We laughed with him. And this is exactly how we laughed with him. So almost under the table because it was so funny. And he made so many pictures of us, so many pictures of everything, that there's proof of his existence, of his role, of his impact, very, very much around us. And I dearly hope it will continue. So goodbye, Amos, but not for that long. And thanks to you for your attention.
Well, thank you, Frederick, for that amazing video and for us to be celebrating the life and the work of Amos. So I'm going to leave you now with Peter. He is a researcher at the Virginia Commonwealth University, and he will have a talk of his work tomorrow, I think.
Hi, welcome everybody. It's my pleasure today to introduce this year's winner of the Biocuration Lifetime Achievement Award. And if we can have the slides for that. As I said, I will not talk about myself. I will talk about Ruth Lovering, our award winner. And Ruth is one of these people who shaped the field of biocuration by her 25 years of engagement in this area. And I don't know if I think I can advance the slides here myself. And before I talk about her, I'd just like to remind everybody that the award is given to people who really had an impact on the field. And these are the past winners of the award. And even if you don't recognize their names, you will almost certainly have used their resources and their work in your daily activities as a biologist or as a curator or as anybody who is interested in biological data, because these guys all worked on areas which are essential, for any biological data you may use in your life. I don't have to go through all of these individually. I just want to point out maybe one or two key aspects, like Chris Mungle, for example, was one of the people who got involved in ontologies, which is one of these key things which not many people know about, but you may just use them. Same for Valerie Wood, who I didn't know much about, but I certainly used her databases such as POMBase and some of the ontology work she did as well. We just heard about Amos. He's one of the key people in the field by founding the SIB and actually some of the databases like Swissprot and Uniprot. Then we have Pascal, who also worked we have a lot of yeast people here apparently, because yeast was essential in the history of biocuration and bioinformatics as well as genomics. Sandra Orchard, you probably have heard of. She founded several databases, and I worked with her on some of them. And finally, we have Ruth Lovering, who did a bunch of things which are shown on the next slide. So she, as I said, has been involved in the field for 25 years. She actually got a degree in genetics from the University of Cambridge with Mike Ashburner, who is the founder of the gene ontology database. And so she's an experimentalist, but she realized apparently that you have to do a lot of bioinformatics work to understand the biology of genes and so on as well. So she got involved in the Human Gene Nomenclature Committee, which is, of course, like the human genome annotation. Gene ontology but also IMEX, which is a database system for protein and protein interaction data. From there, she went on to work on the functional annotation of microRNAs, which is another new hot topic still. She also worked on gene regulation, which is the Greek consortium, and then on functional gene annotation and, of course, the ISB and some of the committees we have. So Ruth's impact is also measurable in terms of citations. You can see her page on Google Scholar, and you can see that her work has been included in a number of different databases, Ensemble, the NCBI gene database, functional annotations, but also patient-relevant databases such as Parkinson's disease data, and all that stuff went into like numerous publications. We have 180 listed here, and more than 20,000 citations. Obviously, she did have a huge impact on the field in general. She also organized some of the biocuration workshops in the past and mentored numerous students and postdocs. And you can also see from that slide that she was involved in ISB for many, many years, and of course, in these committees which I just mentioned. All right, okay, so here are just a couple of citations or quotes from people who worked with her. I don't have to read them, you can read them yourself, because I don't like when people read stuff and you have to read them at the same time. So I just give you 30 seconds for this one. But you can see how influential she was on these people. Rachel Huntley is one of these examples. And then we have a quote from Nicole Vasilevsky, also another person who is involved in the ISP for a long time. And finally, we have Rebecca Fulger, who worked with her in biocuration. I give you a minute to read this. And not to forget, I mean, as you can see at the end here, Ruth told her how to use VLOOKUP, one of these things which I never fully understood, I have to say. But I think it's important, and you should know how to compare lists. That's one of the most important things you can do in biocuration. All right, thank you so much for your contribution, and give us a loud applause for Ruth before we hand over our present for her. So, my goodness. Thank you very much for your contribution, and here is your award.
Thank you, everyone.
So you will do your presentation now, right?
Yes.
Take a picture.
Picture.
And then
Wait,
take
is
a
it
picture.
Okay.
Okay.
So let's take a picture
okay.
first, okay?
I don't know if this is.
Oh, you go.
You're going to fall off the stage.
Okay, yeah. Be careful.
Okay. Great.
Well, I don't think.
Thank you.
Okay. So Ruth will tell us a bit about her work in the following presentation. So I didn't have to explain all the details she did, but you will learn from her directly. Thank you.
Thank you. That was rather overwhelming. And I just want to thank the ISB for all the work they've been doing over the last 20 years or so. And I think it's really important that we all think about how much we're contributing to the scientific community. And it's very easy to overlook that and not recognise our importance. So I think that's great. And I just thank you all, for those of you who voted for me to be given this award, and for those that nominated me. Okay. I first have to remember to say that I've been retired for 2.5 years now. And since retiring, I've gone back to something that I was doing as a teenager, which is lace making. And so there's going to be lots of pictures of lace, because I feel like it's a type of network. And when reflecting on what I've been doing for the last 25 or more years, I realized how important networks and networking has been to myself. And so then I just have thrown in some lace pictures, which maybe will be something else to look at while you're listening to me. Okay, so I'm just giving you an overview of what my talk's going to be about. So I'm going to give the thanks to people to start with, talk about networks and sort of think about my career, the teaching we've taken on, gene ontology and microRNA annotation. So first of all, I'd like to give thanks to the functional annotation team that I led, particularly to these people. So Philippa was the PI for the grant to start with and has been very supportive throughout my career within UCL. Rachel, Barbara, Rebecca and Sharon were particular members of the team, although I'm thankful to all of the team, but they're particular members who really supported me during that time. Obviously, the Gene Ontology Consortium, I'm very grateful to, and particularly Pascal, Val, Julia, and Maria, who really, again, they've extra support that they provided. In addition to other people, I'd like to thank these people. It's been mentioned that Michael Ashburner was my PhD supervisor, but also led gene ontology. Sandra, Colin, Sue, Steve, all these people I'm very grateful to. Without them, I think the work that I did would have been very different. And also I'd like to thank all the co-applicants, collaborators and students that I've had contact with, as well as obviously my family and friends. So career summary, I'm doing this very quickly because my talk has been reduced from 45 minutes to 30, so I didn't want to miss out too much. So my career summary, yeah, in the 80s, I was doing a genetics degree, and then I moved on to doing a genetics PhD using Drosophila. I then did a few postdocs looking at transcription factors, immunodeficiencies, and signaling pathways. And this was looking at human work rather than flies. And then I had a career break, and then I moved on to working with the Hugo Gene Nomenclature Committee. So we were approving the gene symbols for the human genome. And then in 2007, I set up the UCL Functional Gene Annotation Group. And as I say, today I'm now retired. So I retired in 2023. And I just thought I'd show this because I've carried on doing some annotations. The gene ontology has been changing. So there's been a lot of requests to edit the database. And so I've continued to do that. And I think it's quite interesting. You can see, I don't know if there is a pointer on this. I guess there probably is. There we go. So you can see that in 2023, which was the year I retired, I wasn't doing very much annotation. And then in 24 and 25, I was doing a lot more annotation. So those are the updated records. And I think this is because I was no longer writing grants. And I would say that writing grants takes up a lot of time and prevents you from perhaps doing things you'd like to be doing. So what do networks mean to you? And the Cambridge definition talks about a large system. I don't say this out loud now that Peter said people don't like people reading things out, but we've got this large system of many parts that are connected together to allow movement and communication between them. And I think many of you will be thinking probably of computers, networks, and phone networks, and television networks, and things like that. So I'm going to really want to talk a bit about social and professional networks, as well as intracellular signaling networks, which is what I was interested in. So if we think about networking, this is the activity of meeting people who might be useful to you, especially in your job. And they have been so important to me in my life. And I think conferences like this are such an amazing opportunity. And I just want to say to those of you that are less senior, you know, be brave and network. I think it can often be quite scary to go up to somebody that you feel is an important person and to actually have a conversation with them. But you're not going to know how they might help you with your career. And you don't know when that's going to happen, when they might be of use to you. And it's obviously very nice to meet new people too. You don't have to just think about them as being useful to you in the future. But try and be brave and go out there and do that. So I'm going to talk about just a few of the instances where networking has been important, and if without it, my career wouldn't have progressed the way it did. So I started my PhD looking at gene regulation in Drosophila in Sheffield, and I met Michael Ashburner networking at a conference. I actually got to know his students, and then I got to know him at the conference. And while I was in Sheffield, my PhD supervisor had a heart attack and retired, and I had to move to somewhere else to finish my PhD. And because of the networking I'd done with Mike Ashburner, he agreed that I could go and work with him. So without that, I don't know where my PhD would have finished, really. And then I finished my PhD looking at transposable elements, not transposable elements in Cambridge, they were actually in fruit flies. So and then I met Sue Povey, again through a network, which I haven't mentioned here, but I used her, or she was helpful, to get funding to stay at UCL when the Hugo Gene Nomenclature Committee moved to the EBI. And she knew Philippa Talmud and said, this is a very lovely person, you should contact her. She was working in cardiovascular research. And so Philippa supported my British Heart Foundation application, and it enabled me to get funding to stay for 5 years at UCL. And Philippa's networks, she knows so many cardiovascular scientists, and so she then, those cardiovascular scientists were willing to be on our grant applications, and without those scientists, I don't think the British Heart Foundation would have funded the grant. And then I had a friend who was the PA to Steve Humphreys, who was the group head. And she suggested to me that I really should establish a teaching role at UCL if I wanted to stay at UCL. And so I started trying to set up a master's course. And well, I did set up a 10-week master's module, which became a core module for one of their master's programs. And the EBI, who I'd got to know through HGNC, Sandra Alter provided me with lots of material. And so the networks I'd established in the EBI were essential to that 10-week course that I established. And then since then, I've created my own networks. And I've literally written to scientists around the world who have never met me and said, look, we need to get funding for cancer annotation, or we need to get funding for this or that. And would you be willing to be a co-applicant on my disease-focused grant applications? And the majority of times that I've just literally reached out to people that I've never met have said, yes, you know, that's great. It really needs to be done. And we're happy to put our names on those grant applications. Okay, look at my time. Okay, so I'll skip this slide. So I wanted to talk a bit about gene ontology and interaction annotations, and then so the functional gene annotation team that I led, that was our focus. We were focused on human proteins and curating human microRNAs. And the important thing is really that we were focused from a disease side, because we got funding from the British Heart Foundation and from other disease-focused charities. And so we needed to focus on those aspects. So I wanted to just point out the challenges of working in an academic environment versus working in a bioinformatics institute. So in an academic environment, you have lecturers that are core funded and have permanent posts. I actually was given the position of a research associate, so I was grant dependent rather than having a permanent position. So I was always under pressure to get grants in order to stay working at UCL. And to get funding, I needed to demonstrate value, and I needed to explain why being based in the university versus being in a kind of service institute, as they were viewed really, was important. And one of the aspects really was the fact we had direct access to expert scientists. And they could provide us with lists of disease-associated genes. They also contributed to ontology development. They also contributed to our published articles. So we would often, some of those papers were written about the ontology development, but also with scientists that were working in the field contributing to them. And we also gave them feedback to how to interpret their data, because high throughput data analysis was becoming more widespread. Yeah, and also they fed back on the frustrations of using our data to interpret their data. And so when we were applying for grant applications, they were always disease or systems focused. And I think at the time we were the only Go Consortium group that was actually focused on curating disease relevant genes. And I think the other thing to think about, which I think was mentioned earlier, was this lack of understanding by university scientists. And this is quite a challenge when you're a small group in that environment, because many have no idea about what biocuration is, and they have no idea at all about how the data gets into the databases. So we were quite isolated. So I mentioned I did some teaching, so I'll quickly go through that. So we set up a 10-week bioinformatics core module. We've probably taught over 400 students, and we've covered the key gold standard resources. So I've just listed them there. We also had about 30 master students that did 4-month gene ontology annotation projects with us. And 5 of those we got in 1 year because of COVID. We also did these 2-day staff or PhD student bioinformatic courses, and probably taught over 350 staff and graduate students. Again, these were great networking opportunities, because I don't know which way I should be going. I met people who I then asked if they'd be interested in being co-applicants for our disease-focused grant applications. So this is just an example of, this graph is just an example of what we got one of the master students to do. So they were interested in TGF-beta signaling pathways in endocardial cushion development. And so this student focused his annotations on just this 10 or so proteins and looking at their role in endocardial cushion development. And he provided all these new Go annotations. Some of these proteins had never been associated with a heart development Go term, and the annotations he created were much more specific than just heart development. They were to do with endocardial cushion development. So what I've learned from teaching students to annotate is we need to provide them very simple steps to annotation. And I also learned about Jim, who set up this competition called Cacao. And it's a really good way to encourage student engagement, because it's a kind of competition, seeing who can do the most annotations, most correct annotations. And if you're interested in teaching, I would really recommend you looked at this website. The other thing I learned was that students aren't very good at finding suitable papers to curate, and they get very frustrated by the time it takes to find them. And so we ended up realizing that we need to actually give the students papers to curate. And I'm sure that in future, text mining AI will help this. It's essential to have a data to, when they do the curation, you need to know why they've made that annotation. So we asked them to actually provide a description of the data that supported the annotation. Otherwise, it just took such a long time. You had to read the paper maybe 3 or 4 times to work out why they'd made some annotation that was a bit out of left field. And also, because they had to then write these projects up as a thesis to get their degrees, it was really important to have a very clearly defined project with a list of genes to curate. And it meant they wrote a really good thesis. And towards the end of this giving the students project, I realized that the microRNA annotation was a lot easier for them than the protein annotations, because there's a very much more limited type of assays for them to curate. And we provided very clear guidelines for this. And the benefits of projects for students are, we actually sent out a questionnaire to all the students that had done projects with us, and to find out how useful the projects had been. And all of the students stated that these projects had had a very positive impact. They felt the skills were very transferable, and many of them had careers that benefited from understanding bioinformatic resources better. So we had biocurators, but also people that did PhDs, postdocs, there was medical writers and clinical geneticists that that was their careers they went on to. And the projects actually provide a very supportive, active learning environment, and it kind of consolidates their understanding of the bioinformatic resources. And they also appreciated the fact that the Gene Ontology Consortium was willing to put all the students that did projects with us onto their publications, and some of the students' work was actually published separately. Okay, how am I doing on time? I'm running out, okay. So I'm going to do a brief overview of gene ontology because I'm sure many of you know what gene ontology is, but it's a very important resource, and so I'm going to just give you an overview of that. So the aims of the Gene Ontology Consortium is to provide to compile ontologies that describe 3 aspects of molecular biology, which is the function, the biological process, and cellular component, and to enable these terms to be used to curate gene products in any organism. And then they also provide the gene annotation files, and it's a freely available resource. So the idea is that we're going to describe the gene product's biological role. This is just a synapse, so the role of the proteins within this and their location in the cell. And this, I mean, you're all biocurators, so I assume you appreciate the need to provide, a way of grouping genes together so that you've got this idea of what a shared, in this case, it would be a shared biological role and a shared location in a cell. And so for molecular function, we could perhaps describe the insulin receptor as being a protein tyrosine kinase. It binds to insulin, and it also has the term insulin receptor activity. So it's a very singular kind of activity, task, or job. And then the biological process is more complicated, really, and it's this idea of a commonly recognized series of events. So we could describe cell division as being a biological process, or even something as broad as developmental process, such as limb development. And you can expect that signaling pathways will be playing roles within this area. And then for cellular component, we would describe the role of where the gene product is within the cell. So you could say mitochondria, or you could be more specific and say mitochondrial membrane. And as you'd imagine, each Go term has an ID, a name, a nice definition, and synonyms, so that this is, I think, more for curators, so that we can actually make sure we get the right term. And over 3 ontologies, there are more than 39,000 terms at the moment. I mentioned I've been doing a lot of annotation. When I left, there was probably 45,000 terms, and there's been a big reduction in the number of terms, which has led to a lot of more annotation revisions. As you'd imagine, the ontology structure, the gene ontology is not a flat list. The terms are placed within a hierarchy, and so we have multiple parents which are more generalized terms. And then we have children that are more specific terms. So here we've got something like presynaptic organization. And this is a child of synapse organization. So a single term can have multiple parents, and have a term can have multiple child terms too. Okay, so the UCL team was involved in quite a few ontology development projects, and with the funding from the British Heart Foundation, we created over 1,500 Go terms, and over 200 of those described heart development, and 150 described cardiac conduction. We were funded by Alzheimer's Research UK, And this led to the creation of over 180 Go terms. And these kind of describe things such as the blood-brain barrier, synapse, and amyloid processes. So all the kind of processes that are relevant to Alzheimer's. And we were also funded by Parkinson's UK, and this funded the creation of 360 Go terms. And this covered things such as apoptosis, autophagy, and the ERED pathway, again, relevant to Parkinson's. So I've talked about the ontology terms in Go. So the Go annotations themselves enable us to make statements that a gene product has a specific function, it's involved in a particular process, and it's located within a certain cellular component. And this is as described in a published reference and using the results provided by an investigative method. So one of the genes we curated was neurexin 1, and that's here within the synapse. And so it was made a statement that this was a signaling receptor, and it's involved in postsynaptic membrane assembly, and it's located in the presynaptic membrane. Okay, this is just an example of an experiment that was used to create one of the annotations. So if we actually looked at neurexin 1, you can see it's now got 62 distinct Go terms associated with it, which cover a whole range of different Go functions, processes, and its location, as mentioned. And the importance of the gene ontology is that it's being used to enable disease-associated genes to be identified through their shared role or location. And so, for example, when we look at that synapse, the proteins in here that have this red dot, these are all associated with, these genes all have variants that are associated with autism. And you could say that this would give you the suggestion that maybe more of these proteins, I mean, probably obvious, but more of these proteins within the synapse could be associated with autism. But also when people are doing high throughput analysis, genome-wide studies, it means that potentially some of these other genes will be identified because the gene ontology can enable you to lower the statistical threshold by which you can identify the genes that are associated with a specific disease. So in total at the moment, the UCL team has created almost 74,000 annotations to nearly 10,000 distinct gene products. And if we actually look at it by species, as I mentioned, we were focused on human annotation, but we actually ended up annotating the mouse as well. So some human genes haven't been experimentally looked at, and other model organisms are being used to characterize those genes. And often we would then copy the annotations we're associating with a mouse gene to the human gene if they were considered to be orthologs. So the majority of those annotations are to proteins, but you can see we did 9,000 annotations to 800 microRNAs, and also some annotations to complexes. So the British Heart Foundation funded many of these annotations, nearly 40,000 annotations. Alzheimer's Research UK funded 12,000, and Parkinson's funded nearly 7,000. So I don't know what I'm doing. 5 minutes, great. So it was mentioned that we were curating microRNAs. So I'm just going to go over what microRNAs are, because many of you may not have heard of microRNAs, or have heard of it but not really understood what's being described by them. So I'm just going to go through that quickly. So microRNAs are they're non-coding RNAs, and they're seen to often silence the gene expression, and this silencing is happening post-transcriptionally. And so what they do is they bind directly to the messenger RNA. So here's the messenger RNA, and here's the microRNA. And this binding is through near perfect complementarity to the target mRNA. And because of this binding, there's no translation, so you get no protein. So microRNAs are now being identified as associated with disease. They were identified 30 years ago in C. elegans, but most higher eukaryotes have now been identified as having them. And it's thought that the biogenesis, the synthesis of these microRNAs is very tightly regulated. And their dysregulation has been associated with cancer, as well as cardiovascular and neurological diseases. And they're being used clinically as diagnostic biomarkers now, and that research is very much ongoing. And they're also being looked at for therapeutics. So either using mimics of the microRNAs to increase the levels of a specific microRNA, or using anti-microRNAs, which will block the action of a microRNA if it's being overexpressed. So when in 2012, I was writing a grant application, and there were 1,300 articles that needed to be curated to describe human microRNAs, that described human microRNAs. But unfortunately, by the end of the grant, we had nearly 9,000 articles to curate. And you can sort of see this, this increase in research that's been going on with the microRNAs. And now there are more than 25,000 articles that are, if you do a search for human microRNAs and just include one assay, the luciferase assay. So these microRNAs are short. You can see they're 18 to 25 nucleotides long. They have this seed sequence, which is 6 to 7 nucleotides. And it's that sequence that's essentially has to be present, has to align with the target. So that's the key region. But you also have this region up here, which it's less complementarity here. And the problem is that this isn't fully understood as to what exactly, which nucleotides are really important. And yeah, so this is a problem that we don't really know exactly what sequence is required to get this, this match, which will enable the microRNA to target the mRNA. But there are lots of tools available which predict the microRNA targets. So if we look at microRNAs, possibly a third of human mRNAs are being regulated by microRNAs. And microRNAs create this network of regulatory interactions with their targets. And it's thought that there could be between 2,000 and 10,000 human microRNAs. And one of the tools that predicts the interactions is MERWalk. And this predicts them, and it also provides validated microRNA to mRNA interaction data. So if we look at that, we can see that some microRNAs have over 1,000 mRNA targets. So this one microRNA, MER27A3P, is predicted to target over 11,000 mRNAs. Of these, around 300 have been validated. And you can see if you're going to start using an anti-MER or a MER to mimics to add to a human, that knowing that you've got 1,000 mRNA targets means that it's really important to think about what impact that's going to have if you're using that microRNA. Some mRNAs are targeted by multiple microRNAs. So human PPAR gamma is predicted to be the target of over 1,700 microRNAs, although only 6 of these have been validated. So the problem with the prediction tools is that there's inconsistent prediction of the microRNA targets. So if you go to different tools, you'll get different results about what your microRNA is going to target. Because of this problem with the seed sequence and the additional sequences that decide this. So more research is going to be needed to really get that narrowed down so they can be more specific. And on the validated side, different criteria are being used for validated. So the text mining is being a little bit misleading, perhaps. So some of the resources are just using correlation of expression data. So if a microRNA is expressed and at the same time you get a reduction in a protein expression, this is assumed to mean that that microRNA targets the protein. But there could be a network that leads to that change. In addition, you might have a paper where one microRNA has been experimentally validated, but they've actually mentioned multiple microRNAs. And so all of those microRNAs get included in the validated statement in the database. And also there's often a problem with the species information isn't clear, and that might not be identified by the tools. Finally, more recently, people are talking about whether the microRNA itself is expressed at levels in a specific cell that would actually suppress the target gene that the gene that it's targeting, the mRNA that it's targeting. So the curation of, so we were the first group in the gene ontology to curate human microRNAs with experimental data. And so because of this, we looked at developing new ontology terms to enable us to really capture the role of these microRNAs. And we recognized that the luciferase assay at the time was the main approach used to identify microRNAs that targeted the mRNA. And we established new guidelines for the curation, which meant that we had to identify a minimum assay that's needed to say, yes, this microRNA definitely interacts with this mRNA. We also wanted to identify the minimum data to enable us to make a statement about whether the microRNA and the mRNA were interacting within a specific cell or tissue. Also, we wanted to capture downstream target impacts. So some microRNAs, the experiments that they were carried out enabled people to say that perhaps the microRNA had a role in affecting endocardial cell development or something. And we wanted to be able to capture that. And we also identified that because these are such short sequences and the targets were so difficult to predict reliably, we didn't want to just copy data from model organisms and transfer that to the human effective equivalent microRNA, because there might not be true functional orthology between those different species microRNAs. And we came up with this decision table that we got the students to look at. So we got the students to think about this, and you can't see any of these Go terms, so I apologize for that. But we basically gave them a selection of Go terms that they could apply according to the experimental data that was available. So this is a brief annotation example, which basically there's a luciferase assay and a protein levels, and using this data, they would create an annotation. And this is what an annotation might look like. So we have a microRNA here, MER17, which enables mRNA base pairing, and we have the input of the protein that it would, well, the mRNA that it would bind to, and the protein that was reduced expression, had a reduced expression. And then we took this data, so we've got this data that we've created, and this goes into the Go consortium database, annotation database, and then this enables us to make statements that a microRNA is interacting with a specific mRNA. So here we've got BMP2 being interacting with this microRNA. And this data can all be extracted and used to create a network. And so I had some students, 2 students were working on the signaling networks relevant to PAH, which is pulmonary arterial hypertension, which this is a disease where you have an increased blood pressure in the lung arteries. And there have been variants already associated with PAH, which include the BMP receptor 2 and other proteins within the signaling networks within the endothelial cells. So one of the students created this rough interpretation of the data that was available that she picked up at the time. She'd reviewed some of the literature and come up with this. And we gave the students the suggestion that they should prioritize the BMP2, TGF-beta1, and the PPAR gamma networks. And between them, they curated microRNAs that regulated these PAH relevant networks. And this could be represented in this way, where the green spots are the prioritized proteins, The blue are the microRNAs. And while they were doing it, they didn't just curate the interactions with the priority proteins, because we have an approach of doing full paper curation. And so because we're doing full paper curation, if any other proteins were mentioned in the paper and shown to be interacting with microRNAs, then we captured that data too. And there were also long non-coding RNAs that were curated at the same time. The students managed to curate 133 articles relevant to this network and created about 1,000 human gene annotations using 170 different Go terms. They identified 154 microRNA to mRNA targets. So this is more of the data. And all of this data is available through the Go and the Scikit files. You can, if we actually pull that data, reduce that data and just pull out the priority proteins and the interacting proteins, you can actually then, in Cytoscape, you can overlay this with the Go terms themselves. So you can see that Amelia has overlaid this with smooth muscle cell proliferation in green. So any green pie segments are because the protein or the microRNA is associated with that. And then we have regulation of apoptosis or some other things, or regulation of migration. All of these terms are very relevant to the disease phenotype. And you can also see that a single microRNA can regulate the expression of multiple proteins within a network. So here we've got microRNA 21 here is regulating the expression of these proteins that are all associated with this single signaling network. And then, okay, thumbs up, sorry. And then basically Amelia made this more, I think this is more interpretable for people when they're looking at this data. So we've got over 100 microRNAs in this figure, which are targeting receptors within this single, well, there's BMP2 and TGF-beta signaling pathways. And you can see, again, more clearly that a single microRNA can regulate the expression of multiple proteins, and that a single protein, here BMP receptor 2, is being targeted by multiple microRNAs. And I'd just like to thank you all again for your patience and for the award, which was fantastic. Thank you.
You have about 5 minutes for questions, but of course you're also invited to ask questions during the break and during the poster session.
Apologies.
No problem. It's a perfect timing, Ray. We have still 5 minutes. Are there any questions? Do you have a microphone floating around here?
We've got one over here.
Who's taking care of the microphone? Okay. You can start.
Wonderful work and wonderful talk. My interest was in microRNA domain here. So we work on plant microRNAs. And what we have observed is that, especially we work on rice. So for rice, there are 3 to 4 databases which are compiling microRNA genes, and all of them have a different number. And there seems to be a lack of unified curation of the microRNA genes, especially in a species-specific manner. What are your comments on that? How should we handle that?
Well, yes, I would say find a team of students and get them to do your annotations for you.
So, so it's like 5 databases already international. Do we launch a 6th one? Or we do try to find some coherence between what is already there? Because what is happening is every 3 to 4 years, some new database comes up for the same species.
Oh,
But
yes. I don't know. I don't know how you try and standardize. I mean, when I was working at HGNC, we wrote to all the people. Quite often, several people would be cloning the gene at the same time. And we had to write to them and say, you've got to decide on the name. And, you know, you need to contact those people and say, we've already got this database and how can we work together? I mean, Unicron, you know, came together from 3 different protein resources that were all curating proteins at the same time. And they needed to work together. And that's where Uniprot evolved from. but
Yeah.
you need to talk to the people,
Thank you. Yeah.
I guess. Yeah.
Thank you. Hi, Ruth. This is Winnie.
Hi.
I think this is a bit of a fangirl moment and a question. I've had the pleasure of having you as my role model for the last 15 years or so of my own career. So it's fantastic to see you here and very well deserved. I am wondering, as you look back from retirement, what are some of the thoughts that you've given to sustainability of these resources that we have been working for so hard? This is a challenging moment all around the globe, and I wonder if the perspectives that you have gained give us a little bit of thought on how do we make better use and make these resources sustainable with the community.
Yeah, that's great. I mean, I obviously left just before AI was really exploding, which I think was probably a very sensible thing to do, because I would have had to learn so many new skills. And yeah, so I've left that all to you guys. And I think my experience is been the frustration of seeing the gene ontology in particular changing its decisions about how to curate. And that's why I've been doing so much more curation, and also changing the ontology terms themselves and how, and it feels like sometimes the curators need to have more input into the decisions that are being made by the ontology teams and how the databases are being set up. Because I think sometimes, I mean, one of the Go Consortium meetings, apparently there was a discussion about talking about the curators as being kind of like a machine. That's just like, oh, we'll just give the data to the curators. And it's actually, the curators can provide a lot of valuable feedback that I think isn't always recognized, that when you're on the coalface of curating, you really see the problems that are there. And yeah, and I think we're a very valuable community. Let's just shout that out there. Yeah, I don't know if that really answered your question, but you know, that's all I can offer. Yeah.
Thank you. Thank you so much, Ruth. Congratulations for your great career and inspiring path. So I'm not discussing science. I'm discussing, so when you presenting your career summary, you had some kind of, since you get your PhD to get a permanent position at quite a decade with 3 postdocs and 2 years of break. So what will be your recommendation for people, for early career scientists who are a bit struggling with this during this time, you know, a lot of competition, a lack of funds. So what will be your recommendation and suggestion to be resilient, and how? You give a tip that is networking, but any other thoughts regarding resilience and being inspired to continue the career of science?
Yeah, okay. So that's a really challenging one. In Steve Humphrey's lab, he actually had a few people that did sort of internships with him, which I don't necessarily agree with because they're usually unpaid. They're also, he did cover the travel expenses that the people I knew that were doing internships there. And it did give them valuable experience and supported. You know, I think the problem is you send your CV out and it's very difficult to stand above other people. And certainly in the UK, there's a big push to make sure that you can't just, so when I got a job with the Human Genome Nomenclature Committee, someone had said to me, oh, you can probably do naming genes from home. Why don't you contact Sue, because she does naming genes? And so I just contacted her and she said, oh, I've got 3 months money. Why don't you just come and work? Well, you can't do that at UCL anymore. Everything has to be advertised. So you can't just necessarily write and say, I want a job. Can you find, you know, it's not so straightforward. But I do think volunteering has an opportunity to, but make sure it's not forever, but gives you an opportunity to really improve your CV perhaps, so that your CV looks stronger. And I know the EBI has internal apprenticeship opportunities, but it's a very challenging environment. And it's difficult to know where the AI is going and whether it's going to reduce the number of jobs that are available or whether it's actually going to increase them because of the double checking of the AI data. Yeah.
I can say
Yes.
So by the way, so we do have multiple sessions on career options, and maybe Vicky, you can say something about like the times and stuff. And since our break is about to be now, I think we should start the session now with thanking Ruth again. And again, remember that you can ask questions only during the break and during the postal session. Okay, thank you. So maybe, yeah, maybe Vicky can say something.
Great. So on the schedule, there is a panel discussion on discussing career options in biocuration. And as mentioned during lunch, we've got mentorship tables, which Dr. Kutala is organizing. They were mainly for high school learners, but we're definitely happy to expand those to anyone else who might want to sit with a specific mentor, and she can dedicate tables for you. Thank you.
Photo session. Can you say something about that?
Yeah, okay. You will say, yeah. Great.
Can you say something about the photo shoot? No. Yeah. It's in there.
Okay, thanks everyone. We'll head for tea break next door and in the Atlantic foyer, and then we'll be back for the next session, which is methods in biocuration. Thanks everyone. I'll just ask the speakers to move to the front of the thing. Come and take a picture of these people here.
We should ask Ruth.
And how was your trip? It was really good. Yeah.
It was really an interesting trip.
No jet lag.
Well, I suppose, so you said.
Okay, if everybody can make their way in, we're going to get started. So welcome to Cape Town. I am the current interim director for the African Bioinformatics Institute, which is one of the hosts for this conference. And it's really wonderful to see so many people in Cape Town. I promise you the weather will improve, so you will see Table Mountain. I'm very happy to be chairing this first session about new methods and techniques for biocuration. It's really an exciting time. AI has taken over everything, including curation. But even before that, so I was a Swiss procurator in 1999, then an interprocurator. And I think if I had to go back to curate now, I actually wouldn't even know where to start. So I'd have to be taught quite a lot. So it's my absolute pleasure to introduce our first speaker. So I've known Alex for about 25 years, from the days back at the EBI and when he was at Sanger. So Alex did his PhD in the LMB in the University of Cambridge with Cyrus Kothia. And he then joined the Sanger in 1997, and where he started leading the PFAM project, which hopefully most of you know about. He then also introduced the RFAM project. So in addition to his amazing achievements, currently now as head of protein sequence resources at the EBI, Alex has made an enormous contribution to bioinformatics and biocuration. So he's involved in the Wikipedia project to curate entries on Wikipedia. He's been a vice president and board member of the ISCB, International Society for Computational Biology, for a very long time and made major contributions there. He's been executive editors of many bioinformatics journals and is now the editor-in-chief of Advances in Bioinformatics, a really fantastic new upcoming journal. So I won't speak too much about Alex to give him more time to speak. So welcome, Alex, and we look forward to hearing your talk.
Thanks, Nikki. So yeah, it's a real pleasure to be here in Cape Town, my first time here, very exciting. So thank you so much to the organizers for giving me a chance to come here and talk to you today. So today, the title of my talk, The AI Augmented Curator, Ambition, Risk, and the Future of Biological Databases. Today, although I've worked on many different biological databases, I'm going to focus in on the PFAM protein families database, which is perhaps one of the more well-known ones, and tell you about our experiences of using AI in the last couple of years and how it's really changing things on a daily basis, an extremely exciting, one of the most exciting periods, I think, in my scientific career. But this all started back in 1992 when Cyrus Chothia, who later became my PhD supervisor, he wrote this very short paper in Nature, which was essentially a back-of-the-envelope calculation asking quite a simple question: how many families of proteins are there in nature? And the surprising result was that the large majority of proteins come from no more than 1,000 protein families. And this was a big motivator for me. So I like collecting stuff. So I'd collect rocks, fossils, records, make collections of things. And so 1,000 didn't seem that many to me. I could do that. And so started this journey of, while Cyrus wasn't looking, building protein families on the clusters at the LMB in Cambridge. So over the last 25 years, then it's crystallized that my personal scientific goal is to completely and accurately classify all proteins into their families. And I'll tell you a bit about how we're doing with that task. So proteins are built of, we call them domains. And so here we have the molecular structure of a protein. Hopefully you can see that it's got two different domains, the blue-green part made of alpha helices and the yellowy red part made of these beta strands. And these two different domains have different parts of the function of the protein. This is a phospholipase C. It cleaves phospholipids in the membrane. And so we have a zinc-dependent phospholipase domain made of these alpha helices, and then what we call the plat domain, which we believe is involved in localizing these proteins to the cell membrane. So these domains, they form these discrete globular structures in the proteins and carry these units of function of the protein. And nature kind of uses them like Lego bricks. It puts them together in new interesting ways to make new molecular functions. And it's a little difficult to see, but in the bottom right-hand side, we have a range of different representations of proteins which have the plat domain. And this domain is associated with all sorts of different enzymes that are involved in the metabolism, biosynthesis of lipids. Okay, so each of these protein domains, we want to have a record in our database in PFAM. And so how do we make this record? So one of the key elements here is we have, sorry for those folks sat over there that you can't see where I'm pointing, but we have a seed alignment. And this is a set of the protein sequences put into a multiple sequence alignment. We can see which bits are conserved and important for the function. We then use a statistical model called a hidden Markov model, or HMM for short, and that captures the conservation of this protein domain. And then we can use that statistical model to search any set of sequences, our favorite protein database like UniProt, for example. And a curator will pick a threshold such that we think the matches above this threshold are real matches. And then we make something called the full alignment. Then the hard bit sort of starts, which is where the curator has to try and work out, what's the function of this protein domain? And so they go and spend time reading the scientific literature, trying to synthesize the evidence and write a short summary that will be useful to biologists and researchers who are looking at this. Once that's done, then we can create this new PFAM entry and make it available for the public to use. So this is now 30 years since PFAM started, and we're up to release 38.1. It contains over 27,000 protein families. You may notice that this is significantly more than Cyrus Chothia predicted. Okay, it turns out that although the majority of proteins fall into 1,000 families, there's the rest of them, which are much smaller, and there are a lot of them. And so, yeah, I thought this wouldn't take quite as long as it has taken. But I'm hopeful that maybe I can get this done before retirement. So about 77% of all known proteins have a match to one of these domains, and 50% of the individual residues in those proteins, so just over half are covered by these domains. And they allow us to investigate the function and evolution of proteins. If you've got a new virus, you can go and put that through PFAM and maybe understand what the different components are doing. So next I want to talk about how things have changed. In 2021, company DeepMind released this amazing piece of software called AlphaFold, where you could give it the sequence of a protein and it would predict the molecular structure quite accurately. And before that time, honestly, working out where the domains were in proteins was a bit like fumbling around in a dark room. We were sort of guessing where the domains might start and end, and we weren't all that good at it. But so having AlphaFold was like switching the lights on. So here's an entry with a multiple sequence alignment at the bottom of a protein family that I built in 2004. Yes, that's significantly before many of you were born. And it was found in plants, involved in stress response. There's never been a PDB, a structure solved for any of these proteins. And so we just built an alignment of the full length, essentially, of the protein. But then AlphaFold comes along and, oh, actually, the structure of the protein has domains in it. So there are 3 obvious domains. And so, well, great, we can go back to the multiple sequence alignment now and chop it into its domain-sized bits and go ahead and make some new PFAM entries for the individual parts. The first domain is structurally similar to other known DNA binding domains. So it tells us something about what the functions of the individual domains are doing. And if you want to see more, you can go and see the Interpro website and see where all the domains lie in the protein. Okay, so now I want to talk about the excitement of AI and how we're using that to accelerate the development of PFAM. Okay, so here's PFAM growth over the last 20 years. So the number of families that we built in that year is shown on the y-axis here. And each of these is 1 year. Don't worry about the color for now. So in 2024, we were pretty proud of our efforts. So this was because AlphaFold came along. Suddenly, we were able to see where the domains were and build them more accurately. We had the addition of a new curator to the team. And so we passed 2,000 families in 1 year. We were pretty excited by that. And yeah, making things much quicker. So then I want to tell the story of what happened in 2025 and 2026. So in 2025, Christina Rengo's group published this thing called the Encyclopedia of Domains, or TED for short. And so what they had done is taken all of the known AlphaFold models and split them nicely into domains for us. And that's what they're expert at. Before that, we'd been having to go in and open each of these structures and sort of do this manually. It was quite slow. We didn't need to do that anymore. They'd already done it for us. So here we see, we see here a protein, and the blue thing here is the existing PFAM domain, which is this one, the NUDIX domain. And there was a sort of a gap here, which is this domain here. And so we could easily go and build a new entry for that. But then there's still this step of, what does this domain do? Well, so AI has something here. Sorry, this is a bit small. So what's going on here? So I'm a user of Anthropic's Claude. And what I've done is I've given it a paper. This is of the protein structure that had been solved for that protein. And I said, hey, Claude, can you annotate this new PFAM family? It's an N-temrinal, should say terminal domain, found in this paper. It's not the NUDIX domain. And I've also told it, don't search the web, because it goes off and just goes crazy and finds all sorts of rubbish. So Claude goes away, thinks about it, and then it writes one of these annotation files. And it's really good. I mean, really, really good, because it had a good input paper that talks exactly about what this domain does. And yeah, so we started to use this, and this is sort of really helping us to write annotation. The curator's always checking the annotation to make sure the model's not going crazy. So yeah, that was really fantastic. And we also, with the help of AI writing some code, introduced a new script, triage helper. And so we would take the TED domain, go through and make a nice seed alignment through sort of iterative searches. And we could do this over thousands of families. And then this script would sort of triage the list, giving you the biggest ones, perhaps the most interesting ones with information. And we'd add various information, like the species distributions, the way that these domains were combined with other domains in proteins. And then we'd write this, or the program writes this long prompt of all this information, and then the curator cuts and pastes this into Claude, and then the summary gets written. So yeah, this really fantastic. The curator, as I said, reviews all of this and then can make the new PFAM entry. So that was 2025. So what did the results look like? So yeah, we see a big acceleration. So almost 6,000 families were built in that year. And actually now it's worth looking at the colors. So in blue, we have, these are the domains that came from TED, the ones that were chopped already for us. Green ones, these are from another database that's quite like TED called ECOD. And so we'd already been using that for a few years, but TED is kind of better, honestly. So that was 2025, a really exciting year. So 2026, let's see, so we're a few months into 2026, but already AI is accelerating fast and we're running to keep up. So what happens now? So this triage helper, the process has got more complicated. So now we use this thing called Paper Blast from Adam Arkin's group, where they have read the literature and pulled out gene names and associated the sequence of that gene with the paper. And so we can use that to check whether the new family we're building are there any papers published about it? But we go beyond that. We then take the full text of the paper and we ask one of these large language model AIs to read the paper and then make a summary about each of the proteins in that paper. So that now goes into this prompt that we're going to feed to the AI. And also, a sort of a small thing, but really made a big difference is that, so here's part of the interface that the curator sees. And rather than having to cut and paste this text into their AI, now they can just type L and then this gets sent off to ChatGPT 5.4 now via something called OpenRouter. And you can just choose whichever model you want to use. So this really smooths the process. Again, the curator's always reviewing the annotation that comes out. And so what are the results of all of this? Big reveal. Ta-da! Okay, so we are in 2026, and actually we've already exceeded where we got to in 2025. So if we carry on at the rate we're at, we'll build 24,000 new families in 2026. That's basically more than we built in the previous 25 years. Who knew? I mean, maybe I should have just waited till now and then started. I could have done something really useful in the last 25 years, but never mind. So there we are. And I really sort of wondered, could we ever finish the classification? And I thought, really, no, we'll be going and it will go for another 30 years beyond my retirement. But now actually I'm thinking, oh no, actually we may be able to complete this classification, actually complete it in the next 2 or 3 years. It's really phenomenal. Okay, I have no idea how I'm doing for time. Oh, I have 5 minutes. Okay, we're good. Is that really 5 minutes, or is that 5 minutes till the end of questions? Okay, I have 43 seconds. It's no problem. So I wanted to say something about vibe coding, because I think this is pretty interesting for our community. So vibe coding means writing essentially in English or whatever your favorite language is to your AI to say, can you write the program to do this thing? And I was doing this last year and it was a bit of a pain, kept making errors. You had to loop around sort of 10 times to get the thing to work. But in December, the new generation of models got insanely good at doing this. And more or less, it would just write the code to do the thing first time, maybe a few little tweaks. And I haven't looked at a line of code in about a year. I've written more lines of code in the last year than I have in the rest of my career, I would say. And many of the developments that took a few days would have taken months for a software developer to develop. And so here's just a sort of a vignette of one. Each of these boxes is a different PFAM family, and we're looking at the relationships between them. Green is good. They're things which are together in one PFAM superfamily. We call them a clan. And this visualization really makes it much easier for us to kind of work out what should be added, what should be taken away, and so on. So yeah, vibe coding, really fantastic. And it turns out actually my curation team are now starting to do this too. They're developing software that we're using every day in these pipelines. And they bring their expertise of biology and the curation problem to solve these problems in a way that software developers just kind of miss the point sometimes. I wanted to mention that also we're using the AI to do security audits of our code because we make it all available on GitHub. And the AI, there's sort of some concerns that AI can be used to, hack your databases and so on. So if you're running a molecular biology database that's made publicly available on the web, you might want to do this now before the next generation of models comes in. We have a little window to make our systems more secure. And so one of the dreams I had, so we have the PFAM database is a 25-year-old legacy Perl code base. And I had this dream, please rewrite the entire PFAM code base in Python. Well, about 3 weeks ago, it was a bit more complicated than this prompt, but I did that. And it converted into about 22,000 lines of Python, and it took 1 day. Incredible, just incredible what can be done now. So although I'm hopelessly over time now, sorry, Nikki, there may be fewer questions, but I'm here the whole time, so you can ask them in the breaks. So I wanted to say something about the risks. And so I liken this to Scandinavian smorgasbord. There's so many AI risks and issues. We could spend an hour or two just talking about these. AIs can hallucinate, they can make things up. And so you have to be quite careful to, the way we do this is we just give the AI the information that we think is good and say, don't look anywhere else, use this information. But it's worth noting that, humans hallucinate and none of us are perfect. So, just because the AI isn't perfect is not a good reason not to use it. Is it important to track AI versus human annotations? Probably is. Do we have a standard system for doing that? Maybe we do. Someone should tell me about that because I'm not sure how to do it. In PFAM, actually, we don't label the AI curation because we carefully review it all. So that's the decision we took for that database. Maybe for others, we'll take different routes. I'm worried about the cost of AI, both sort of in terms of money, but also environmentally. Building all these data centers everywhere, consuming vast amounts of power is not good for the planet. Issues with intellectual property. There are existential threats. Are we going to be taken over by swarms of robots? Oh my God. Not much I can do about that one right now, except build a bunker and fill it with food and maybe a shotgun. Could it be useful? I'm not sure. Will biocurators become redundant? No. Actually, I think curators will become more and more important. The AI feeds off of data. You need good data to make good AI. And actually, what I think this does is that, within PFAM, our ambitions have just got bigger and bigger and bigger and bigger. And then, okay, now I finished PFAM, what do I do next? Well, we don't know what the functions of all these proteins are. Maybe I'll go solve that problem next. So I think it's really exciting times, and you have to run to keep up, but it's definitely worth doing. Okay, so in conclusion, AI is massively accelerating what we're doing in PFAM through development of code, but also summarizing the literature and helping augment the curator's work. And vibe coding is opening up this amazing opportunity that we can all be software developers. Fantastic. So and finally, I think we can all increase our ambitions, do more with the resources that we have. And I'll finish off by saying a big thank you to all the team that made this possible and you for your attention. Thank you.
So we're going to put Alex on the panel at the end so that if you have a question for him, you can save it for that. So I'm going to hand over to my co-chair, Stephen Ariemo, who's going to introduce the next two speakers.
Thank you. Thank you. Good day, everyone. So our next presenter is Mr. Mugumi Atwin from African Center of Excellence in Bioinformatics, Uganda. He will be presenting about the LWAZ platform, AI-assisted data harmonization and metadata curation for multi-site health research in Africa. Ladies and gentlemen, let's welcome Mr. Atwin.
Good morning to you all. Good morning to you all. Yeah, it's not easy being on stage. So, all right. So I'm going to present to you our approach. So we've been doing a lot of work, and over time we have had to pivot in several areas. So I'm going to just present to you some of the work that we've been doing under LWAZ, the Open Data Science Platform. Okay. All right, so in the DSI Africa Consortium, we have so many research projects, as you can see there. This is just to help you build a picture on our reach. So we have several countries with several projects and so many data sets being collected all over the continent. And obviously, as we start to go deeper to look at what kind of data they are dealing with, we run into a couple of problems. And I know we have talked about several of these. They are going to be iterative. And one of them is how do we get this data to work together? How do we harmonize it? How do we make it possible so that people on one side of the continent will be able to work with people on the other side of the continent? So these are the kinds of issues that we are trying to sort out, to make sure that everybody in Africa is able to find a way to work together and thereby promote research. And the other issue, of course, was how do we find this data? Because there are policies involved. There's all sorts of things that you need to go through in order to get data. Of course, PIs also, they have their own thing going on. So we had to figure out a way to, and this was really a cumbersome process. I know that it's just one slide, but it represents many, many nights of tears and follow up. So the idea we came up with was to come up with a metadata catalog through which we could actually find a way to know who is doing what. So the catalog just gives you an address to where the data is. We are not actually capturing the real data itself. It's just a pointer to something. Okay. So the way the catalog is built, of course, we have somebody, we employed what we call metadata stewards, and the catalog has a REDCap back end. So the metadata steward is responsible for their organization. They input the data into a REDCap back end, and then we have an API that is syncing this into a front end where people will be able to search for data within the LYZ ecosystem. Okay, so in the catalog right now, we have several types of data. We have genomics data, we have mobility, air quality, clinical pathogen data. So there are so many things that we have been able to achieve and bring several groups together. So if you can, when you get a chance to look at it, you should be able to find a lot of this information. So yeah, this is what I was talking about earlier. So we have metadata stewards. So every project has one representative, okay, who is responsible for whatever goes into the system. So we have a catalog team where we, when we interact with these projects, this catalog team is responsible for helping onboarding these different organizations. So the steward submits their metadata, and they're also responsible for making sure it's updated from time to time. Okay, so there is a feedback loop. Before all the data is published online, we have to have several calls to make sure that we are accurately representing what they gave us. Okay, so then after that, we have the catalog go line. So there have been multiple iterations of what the catalog looks like. It looks way better right now than it did a few years ago, obviously, and we are still improving it from time to time. Okay, and of course, there are standards that we had to plug in to make the catalog interoperable. So the dual codes, the data connect and passport. So we tried as much as possible to incorporate this so that it's possible for it to connect to other existing databases. So this is just like a rough picture of what it looks like today. We have over 100 plus data sets from 22 countries and multiple domains. It's not exactly what it looks like, just a representation, but the major point is what you're seeing at the bottom. This is actually a lot of work has gone into making this happen. Of course, the other issue I talked about was harmonization. How did we get the variables to work together? We are having issues like inconsistencies in variable naming, missing metadata, and then, of course, the fair compliance gap, data sovereignty constraints. So these are some of the issues we had to deal with in order to figure out how to get the catalog working. And as a result, to answer that question, we had to come up with a metadata harmonization tool that helped people put things together. So the idea is you want to map a data set to a certain other data set that you have, and you want to know if the variables are similar. So there are different stumbling blocks we run into when we try to use online mechanisms or LLMs there were issues with data sovereignty. So we decided to employ, we decided to be creative and deploy LLMs locally. So the metadata harmonization tool that we are working with actually runs locally on your machines, and it's available on GitHub on that link there. So it's AI powered. You can also download a Docker deployment for that. So the reason why it's local is because people are not comfortable. Of course, health data is sensitive. People are not comfortable to let some of their data go online. So we had to figure out a way to make it local so that everybody is able to do their data, analysis comfortably. All right, so I can see my time is running out. So the idea of the metadata harmonization tool is you have a code book, and then you have another data set that you want to map it to. So we used a technique called RAG, which is retrieval augmented generation. So we give the LLM both the source and the destination, and then it is able to map for you which variables are appearing in which data set, which ones match. Okay, so the recommendation are then presented to a human annotator. So it's not the AI that's doing everything. The human annotator gets to see what the AI does. So it's a recommendation, it's like a recommender system. Then at the end of the day, you're able to say, maybe you got this right, you got this wrong, and then do another review. The other thing that it can do is that it is able to transform data. If you have like categorical data, numerical data, you'd be able to transform it. We have improved it to a level where now it's like an end-to-end system. You can put in your data set and then have a result pushed out, all done locally on your machine without having to use Claude or ChatGPT or anything. Yes, so that's how the AI works. We get the study protocols, we chunk them into vector embeddings, we add a large language model to it, and then get the variable descriptions. In the right corner, you're seeing what the output would look like. So it would tell you like variable A matches variable X, and then it gives you a certain percentage on how confident it is. Then you'll be able, as an annotator, to tell if it's actually correct or not. So these recommendations vary based on the kind of model that you're working with. So we found that smaller quantized models normally hallucinate on the kinds of instructions you need to give the model to do this situation. So if you want better results, you'd probably have to think around having more capacity in terms of infrastructure so that you'll be able to host bigger models to help you do. So this helps save a lot of time. So yes, it's not the LLM here. I'm trying to say it's not the LLM only that's giving the final decision. You have a human in the loop to help you to validate the process. And we designed it for privacy because we wanted people to be comfortable to use it. So it can all be locally hosted on your machine without any cloud infrastructure involved. So we have tried it. We have done tests on a couple of hubs and gotten some feedback. And a lot of the people are appreciating the effort. Trying to rush through this. So ideally, the complete ecosystem now looks like this. We've discovered the data, then we standardize it, harmonize it, and then we get into analyzing it for research purposes. Lessons learned, invest in human infrastructure. There's definitely a need to get these systems up and running. The processes, like the former presenter said, there's a steep curve and growth in how these softwares are changing. But the kind of human capacity that is needed to put these components together is what is lacking. So if you're very used to just using ChatGPT and all this, you might not know what's happening in the background, and we need to, yeah. Thank you.
Thank you so much for that amazing presentation, Mr. Mugumi Atwin. I'm sure there will be quite a number of questions on LWASI, and it will be answered in the panel discussion. Without wasting much of our time, I would like to invite the next presenter. So I'll introduce Dr. Anissa Agura. She's from University of Mauritius, and she'll be talking to us about classifying African and non-African genetic studies in PubMed, is NLP techniques. Let's welcome, please.
Hello, everyone. So I'm Anissa Agura from the University of Mauritius, and I'm also a member of the AfriGen-D, the African Genomics Data Hub project. Okay, so this is the first presentation that is mentioning the African Genomics Data Hub, at least for today. So quickly, so this is a project that aims to develop resources, tools, ontologies, standards for use by the African continents to be useful to describe the African genotypes, genomics, human genomics. Okay, so as part of this project, we have the. Let me look here. So as part of the African Genomics Data Hub, we have the genotype to phenotype project. So what is the aim of this project is to build a resource that will catalog the African-specific genotype to phenotype associations, okay? So the very first and perhaps the most important step would be the data curation. So after selecting, so this is the first part on this slide in the process. So after selecting the disease and all, so we need to identify human genomic studies describing or relevant to the African cohorts. Okay, so this is the step that I will be focusing on. Okay, of course, after we have done this, the next step, the curators will dive into the different publication and extract the genotype and phenotypic information. But me, I want to just focus on the selecting the publications that describe African variants. Okay, so to do this step, so it's very simple step to describe, very easy. We have publications, we just want to know whether or not they are describing African. So we know manual curation it takes time, and it quickly gets difficult when we have an increasing number of publications, just like Dr. Bittman says. So we need to have recourse to computational techniques, and we already have many text mining tools out there, even under the NCBI, but as far as we are aware, none of them explicitly tells you, oh, this publication will describe this population. Okay, so we have a very simple problem here, at least easy to describe. So what we are looking for is to have an automated system that will take a PubMed ID or a publication and will tell us whether it will report African variants. Okay, so because here we are dealing with publications, we are dealing with natural language, so we may not achieve the 100% accuracy, but at least it will be the first step in this one. It will help to get a first pass at the screening of the publications. Okay, so this is the aim. So we've done something with NLP. Okay, so we've done something with the natural language processing. So we are able to extract demographic related terms and from the PubMed ID, thanks to using the NCBI utilities to extract to retrieve the full text articles, and we use Spacey. Of course, Spacey is one of the NLP tools, so there are many out there, and now many are integrating AI. So we are investigating the AI also as well. So when the full texts are available, so our system performs pretty well, so with 80.6% accuracy. So quickly, I will walk you through the methodology. So we all know that there exists many NLP tools out there, and now increasingly many are integrating AI. So we, for example, Hugging Face. But at the time that we did the work, we chose Spacey because this was it is a Python library, so many now we know that Python is the language to use for bioinformatics. So it's open source, the Python library, and also it's very fast because here we are not dealing with small comments. We have full text publications that we need to pass. So for all these reasons, so that is why we use the Spacey. So, and also Spacey will provide you with the basic NLP techniques that, you know, any NLP software should have. For example, tokenization, which will break your text into sentences, removing words that carry little meaning, and we have the stemming and lemmatization. So for those who are not familiar, so when you have a word, so stemming will just get you the root word. For example, running will be run. Lemmatization, this is a little bit more sophisticated than stemming. For example, for better, it would not be bet, but it will be good. Okay, so using dictionary definition to get this. And also we have the part of speech tagging, which will take your sentences and categorize your the words into verbs, nouns, etc. And there are other features as well. So this, in brief, this is our NLP pipeline. Okay, so I will quickly go through each of these steps. And so the first one, we needed a working data set. So we took 1,000 random PubMed IDs, in particular those that have full text. So what do we mean by full text here? It's those that, in addition to the title, the abstract, the keywords, we have the body sections. Okay, so now that we have these text, so which sections are we going to target in these papers? Because not all papers has the same structure, so they use different section titles, subsections. So we need to know which of these. Okay, so if, for example, I know if the demographic term is mentioned in the title, that is straightforward case. Okay, as you will see later on. So this will be that straightforward case, but often it's not mentioned in the title. So we have to go into the section of the body. So for this, we had to target what I will refer to as the methodology and results. Okay, so using NLP, using the SPACY, we were able to identify all the sections and subsection title, and then we keep, okay, so this is after manual inspection, we keep only the 20 most frequent nouns, which would represent the methods and results. So for example, you would see here, for example, we have the materials and method, we have the experimental design, we would have, for example, participant recruitment, we would have all these terms there. So now that we have the sections that we want to target in the papers, so we need the terms that we are interested, the demographic terms. So now this is very flexible. For this work, we are using the Python library. Again, Python, we are using the PyCountry, but this could be not replaced, but this could be augmented in addition to the list of countries provided by the PyCountry. We could also use what we have heard in the last 2 days, the AFPO, any ontologies that is describing ethnicities, population, etc. Okay, so in addition to the countries here, to come back to the work, in addition to describing the countries, we also added, terms that would be used to describe, for example, African cohorts. So we will have Africa, we will have East Africa, West Africa. So similar terminology. Okay, so now that we have our demographic terms. So now we need to target those sentences that is giving us these demographic terms. Okay, so again here we use NLP. So and then we will get a long list of sentences. So we had these number of sentences from the abstract and from the method and result. So this step, the next step now was to manually curate this as positive and negative. So what do we mean by positive? So we have an example here. So for example, our findings may improve early recognition of PD characteristics in Chinese patients. So that would be, for example, a positive sentence, because this is telling us that this is Chinese cohort. But a negative one would be, for example, we perform our analysis on dry blood spot by tandem mass spectrometry according to Italian guidelines. So they are reusing the Italian guidelines. This has nothing to do with the Italian cohorts here. So these are examples. So again, this step here is manually curated, but as we have seen, so we are developing confidence in the using AI technology. This could be, again, augmented using maybe as a first pass the AI and then move on to the manual curation. So we were able now of following this step and choosing, of course, a certain threshold. So we were able to identify the nouns that we will use for human participants. So these were, for example, patient, participant, etc. And we have similar thing for the verbs. Okay, so in NLP, these are the bits and pieces that you need. Okay, so you need the verbs and nouns, the terms that you're interested with. So having done this, so now we are able to, see whether what we have identified, whether it, to evaluate it, okay? So for this, we use the African Genomics AGMP project. So what is the AGMP project? It's the African Genomics Medicine Portal. So this group, they did a great job of curating over 1,300 PubMed IDs. So these were manually curated, and we know for sure these report African variants, okay? But not all of these publications have full text. So these are the number of full texts that we have and the number of abstract only. So this would be considered as our positive data set. So we need a similar one, but for non-African or negative data sets. So for that, we use the GWAS catalog. So in GWAS, so for those who are familiar, it does report the populations. So we choose those that have no mention of Africa. Okay, same thing. So we will have some that with full text and others that with the others without the full text. Okay, so now that we have the data set, so this is in summary, but what is important here is number 6 here that I want to emphasize is that when we have the demographic term in the title, we can say for sure that we have the paper can be classified according to this. Otherwise, if it's not, we go down priority. We will go down to whether it is found in the method and articles, and then finally some. Okay, so we are nearly there. So please here, I want you to focus on the first 2 sections. So we have the full text data set. So if you have a look at the diagonal, so we look at the true positive and true negatives, and we see that we have high number. So the issue comes with the, I'm going to very fast. This is crazy. So we have a high number of unclassified. We have a high number of unclassified publication when we are not using the full text, and that is why we need to consider other techniques. So we need to have a look at NLP tools that integrate the AI, and also look at the LLMs and investigate things like the ChatGPT, DeepSeek, and also, but we need to know the cost and everything. And also, maybe later on, we will ask a question to Dr. Bateman. So that's it. That was great.
Sorry to rush you, but we do have to stick to schedule. So, but you'll have an opportunity to get questions in the panel session. So our next speaker is Dr. Barbara Stradzel, sorry, who's going to be talking about refining bioassay metadata in Campbell.
It's a complicated surname. So I'm actually Austrian. I wasn't on the list. I work for the EBI, but I'm Austrian. But this is a Czech name, so don't ask me how it's pronounced. So I want to talk about the Campbell database and our recent efforts to better annotate bioassays with metadata to make it more AI ready. How do I go forward? Yeah. So actually our team, the Chemical Biology Services team, we look after 6 different services. I don't have time to go into detail, but Sure Campbell, our patent resource, fully automated, Unichem, a cross-linking system between different chemicals, widely used among the EBI and outside. CABI is like an ontology and the database for chemicals of biological interest, and very new is OPSIN, a tool developed by Cambridge University, but we host it and maintain it, which converts UPAC names into structures. But today I only have time to focus on Campbell. So what is Campbell? It's a drug discovery database. So basically what it depicts, like in very simple terms, how a small molecule or a drug-like molecule interacts with different targets. And this interaction is being described by the bioassay setup, so to say. So it really stores experimental data. And in this talk, I really want to focus how we want to improve annotations for these bioassays to make them more machine readable, AI ready. I have to mention, so Campbell is now 16, 17 years old, so it has been fair from the start. I'm not mentioning fair on any of the slides in this presentation. So, but this is really to keep up with the needs to produce ML-ready data. And this is really the case that, so a lot of drug discovery scientists, also all big pharmaceutical companies are using Campbell data. And this is because it basically depicts 50 years worth of data. So we extract data from scientific literature, from 7 core medchem journals. And the other large branch of data is coming from deposited data sets, also neglected tropical disease information that we host in Campbell from the start. We got such depositions. And so really, if you go back in time, there are publications in Campbell starting in the late 90s, 70s. So it really provides this cross-target biological context, which is really valuable for drug discovery. Yeah. So looking at what is a bioassay that you probably all know, how can we actually describe a bioassay? And you can basically split this into several important pieces. You need an identity, assay identity, the description, which I will speak about quite a lot in this talk. So a free text summary of the experiment. And we want to make use of this information now more. Then the assay type, is it a binding, functional, ADME assay, a physical, chemical, toxicity, and so on. The biological target, of course, the assay format, like assay organism, cell line, and the actual readout. Yes, and of course, also data provenance is important. So where does the data come from? And so this is the free text assay description that I've spoken about, just one very simple example. So you can already see in this example, it's very easy to extract certain entities and annotate them. And we actually do this since many, many years. But now we're trying to improve it. And so we have realized, sorry, the format did look different when I looked at it. So that there is actually a template that our data extractors are using. So when they write this assay description, they use this template. You can see that there are many things that could be better annotated by using this template, right? And so in the talk, I will focus a little bit on how we now extract experimental method and how we extract the dose unit, route of administration, and so on from this assay description. Yeah, so this is an example for an experimental method. So very briefly on data curation. So my team is like 5 people, 4 of which are curators, 1 chemistry, 2 biological curators, and 1 person just doing drug data curation. And so I don't have time to go into detail, but we very thoroughly annotate targets in Campbell and assays, but we do also standardize chemical compounds following EUPAC rules. This is a fully a process that is published on GitHub, and many other companies are using it as well. But now I really want to focus on, as I said, target assignment and how we can better make use of assay descriptions. So how do we do the target assignment? So it's quite tempting to think of a target as a protein, but it's not so often the case in Campbell. So actually 2/3 of our bioactivity data, there the target isn't a protein target, but it's something like a cell line, a whole organism, or a tissue, for instance. So how the curators work is actually they would first assign the target type. So, and accordingly, then we have 7 different notebooks, and then the curation is done semi-automatically for each target type. New targets are being assigned, and ontologies are being mapped. And we also worked on enhancing our data checks that we do to increase the data quality. Sorry. And so now about assay annotation and how we can improve this. So one thing that is there since a few years is the assay format and assay endpoint. So this uses the bioassay ontology. So also from the assay description, basically, using a rule-based approach, we would annotate this bio format. So this is something like an experimental system or a biological context in which the experiment has been performed. And you can see some examples. So something like a tissue-based format, a cell-based format. The other thing is the assay endpoint. So that's the specific biological or chemical quantity measured. So we do annotate things like IC50, EC50 with a bio term as well. And then there is the assay type that I mentioned already. So is it a binding functional assay and so on. And we wanted to go further from there. So we basically worked on 3 different things in the past 1 or 2 years. So one is improving metadata for ADME assays. And there we also use this assay description that I mentioned before. So this was a regular expression-based approach. We would use it to annotate the dose unit, and route of administration and time. And all this information is now available in Campbell via specific metadata tables. And that really helps you to identify assay data that you might be interested in, right? So in machine learning, the problem often is that you are interested in a particular assay, but it's hard to identify similar assays that you can use to combine your data for. And that is a piece of work we published recently in Journal of Cheminformatics. So here also we have been using named entity recognition and NLP. And I have to say, this was before this big hype in LLMs now came up. So this was really a side project. We started to work on this 2 years ago. And at that point, you couldn't use ChatGPT. Maybe now I would do it differently, but it kind of worked. So what we did is we developed 2 models. So one named entity recognition model to extract the experimental methods from the assay description. And the other thing was a multi-class classification model to annotate the broad assay aim of an assay. And how did we do that? And I think that's maybe the most important slide of this talk. So I want to emphasize also what Alex Bateman said. I think biocurators are more important these days. And so we really made use of them. So altogether, 2 people in my team, they annotated more than 1,000 assays manually to be able to have a really good gold standard data set. And so on the left-hand side, you can see how we annotated the experimental method. So we've chosen only binding and functional assay for this first round because that was easier, and we focused on literature data. And so they would annotate the experimental method, and then we would train a model on the basis of that, do some cross-validation. We even did some external validation in the end by picking random assays and checking them manually, like 200. But we also, and this is what I want to emphasize, also for the mapping to the ontology, we did some benchmarking. So they also looked into, how many was it? I think 400 or so mappings to the bioassay ontology and manually annotated it. And here, really, this was the rate limiting step. So it was quite easy to identify the experimental method, but we could only map around 20% of these terms to the ontology. It was much easier for the classification model because this was basically just 7 classes. So annotating them with an ontological term was quite easy for the model. Yeah, here are some results. So you can see some examples on the left-hand side, the experimental method, and an example of how this nicely maps to a term. But as I said, it wasn't always such a clear case. The tool we used for ontological mapping is called text-to-term, so it's based on semantic similarity. But we are thinking now of moving to a dictionary-based approach just for the mapping, because I think that is a good investment, like a few annotators basically providing a dictionary, then keeping it up to date, and then you can have more reliable mappings, I believe. And on the right-hand side, you can see the 7 new SAM categories that we have been introducing. It's not available in Campbell yet, but hopefully very soon. But you can find all the data as a supplement to the paper. So, and this is how we see this working with the existing tools, like in a modular way. So we already had the assay format annotated since, I think, maybe 10 years. And now we are also making available the broad assay category and assay method. And as I said before, this makes it much easier for a user to select the right assay and to select the assays that they want to combine in order to build a larger data set for instance, machine learning purposes. And this is a little bit of a vision. So we do actually already annotate a lot of these entities that I'm showing here, like cell lines, tissues, organisms, and map them to different ontologies. We're also using Cellosaurus, for instance. But to go further and making more use of this assay description, and then ultimately even going to full text and extract this information from papers would be even better, I think, to make the data more reproducible, interoperable, and then finally developing something like an assay classification. So where you could then really increase findability. So you could then say, how can I find and extract all bacterial antivirulence assays in Campbell? This is the team, so it's a large team. As I said, we are looking after 5 different services. And I want to point out the person on the left-hand side, second row, is Emma. She is the senior curator in the team. She does amazing work, so I wanted to point her out. She was really very important for all this work that we did here. I'm happy to answer questions if we have time.
Thank you, Dr. Barbara, for taking us through Kembu. I would like to introduce the last presenter for this session, Ms. Blessing Oselu. She's from Covenant University, Nigeria, and she's also the president of International Society for Computational Biology, Nigeria region. She'll be talking about Transcest PRS doc. I welcome Ms. Blessing Oselu. Please welcome.
Thank you. Good day, everyone. I'm Chioma Blessing Oselu from Covenant University. I'm here to make a presentation on my PhD work, Transcest PRS, a framework for evaluating multi-ancestry polygenic risk scoring methods. So I'll be using this outline. So as opposed to Mendelian diseases where, one gene or genetic variant is responsible for the outcome of the diseases, polygenic risk score is a single value estimate of an individual's genetic risk. So basically, scientists thought of a way of, mostly for complex diseases, so as opposed to just one variant causing the disease, different variants spread across the genome, putting it together into a single value estimate and terming that single value estimate the polygenic risk score. So this is the formula for calculating polygenic risk scores, and it has quite awesome applications in clinical translation, which includes disease risk prediction. So it's also being used in stratifying patients into different risk tiers, giving low risk and high risk, so that in the clinic they can propose some early interventions for those who are at high risk, and also for tailoring treatments and fast early screening strategies to those. So there is a problem for polygenic risk scores. Now, these models have been trained mostly on European GWAS, and now it's quite obvious why it loses most of its predictive power when applied to African and other underrepresented populations. And now there's a new advancement called multi-ancestry polygenic risk scores to solve some of these problems. But now the thing is, there's a large family diverse groups and classes of multi-ancestry PRS tools ranging from different statistical classes. I always call them different flavors of multi-ancestry tools. Some of them belong to the Bayesian class, some of them belong to the machine learning class. And the image to the right shows the different multi-ancestry tools included in Transcest PRS. So, but now the thing is, even with these multi-ancestry tools, users are not sure of which to choose for their data sets, as well as these different tools have different input formats and dependencies involved, which means that you have to go to the GitHub of these respective authors to download them and use them. And then depending on how you want to use the polygenic risk score, sometimes it takes a lot of time. And this is why we developed Transcest PRS. So with Transcest PRS, our solid aim was to develop a reproducible framework for benchmarking multi-ancestry PRS methods and make it accessible via a Nextflow pipeline and a web application. So first, we sought to containerize the different multi-ancestry tools, then we orchestrated with Nextflow pipeline, then we benchmarked them across computational memory and wall time, and then we also integrated a biological interpretability layer and then deployed as a fully accessible web application. So this is our interesting and beautiful Transset PRS framework, beginning from the pre-processing steps to PRS construction, using the different flavors of the multi-ancestry tools depending on the user's interest, and then evaluation. Currently, we have integrated the AUC, that's the area under the receiver operator curve. Then we have the Nager-Kirk R-square, liability scale R-square, and then the R-square for quantitative traits. And then the final module, which is our major contribution, the biointerpretability layer, which involves the SNP level, gene level, and pathway level, because we want to be able to tell, the biological or functional annotation of or interpretation of the different outputs of these tools. So beginning from the first objective, which is the containerization of the PRS tools, this is just one slide, but it took me about a year and a half of my PhD to do this. Yeah, so I had to go into the different GitHub pages of the authors to pull the dependencies together into Docker images. I learned a lot. It was a season of patience, lots of it. So thanks to my awesome supervisors, it was quite easy to reach these authors, because by the time I sent emails and I copied them into the emails, they respond to me faster. So after that, we also containerize our preprocessing steps into Docker images so that it's easier for users to, they just pull the images and they're able to preprocess. So we have 11 tools and 22 images, and all of these are hosted on our GitHub for anyone who wants to use it. So we also integrated the quality control pipeline for the different input data sets, because, the quality of our input is what determines the quality of our output. So there are different stages involved here. Then we tested on prostate cancer data sets. We extracted data set from GWAS catalog using the following GWAS catalog IDs, and then the database of genotype and phenotype for the target genotype phenotype data. And these are the different sample sizes. So also we developed we designed a validation protocol to test or to see how what was the word to use to validate the equivalence of our containerized PRS tools with the manual installation via the GitHub. And then we used the secure hash protocol to do that, and then Pearson correlation. And we found that most of them had about above 0.94 correlation with the manual installations, which was really good. Then we also, integrated it using Nextflow. We chose Nextflow because Nextflow has a very good community of users and support, actually. And then it does this cool thing where when you're done, you're running the pipeline, it has a built-in profiling where it captures the CPU, the peak RSS, the wall time, and the input or output for every task. And then it gives you the different graphics, the different plots for it, as opposed to just, writing scripts on your own. So Nextflow is really cool for that. And then we also sought to benchmark across the different tools using this data set. PRS CSX are coming first, topping every other Bayesian family, and then TLPRS, which is the transfer learning PRS, ranking last. Then the compute costs across the different tools with the Bayesian families, having about 2.8 to 4.5 hours taken to run. And then their Bridge PRS and XPARS, they burn RAM more than the other tools. But C++, the clumping and thresholding approach were fast and light. And this explains why in most polygenic risk score studies, they use this C++ approach, because it's fast and it's easier to execute. Then the fourth objective is the biointerpretability layer. So our main motivation for this is because AUC tells us helps us to discriminate between cases and control, but it doesn't tell us which SNPs each tool picked. It doesn't tell us whether those SNPs fall in genes or intergenic regions or regulatory elements. It also doesn't tell us whether those genes are known disease drivers, and as well as the pathways of those genes. And so this is one thing that we added to Transset PRS. And from this plot, we can see that CASC19, PCAT, and GALNT-L5 were all shown from the chromosome 8 at the long arm 24, and this chromosome 8q24 is the most replicated prostate cancer locus according to literature, and this shows that most of the genes discovered by the tool is actually has a biological signal. So this slide shows Venn diagram showing the SNP overlap and the gene overlap. So one SNP was shared across all 4 tools. So we picked 4 tools across the 11 tools. So it showed that one SNP was shared, and then 4 genes were actually picked by every tool. So this slide now shows the functional annotation, giving the variants, whether they're coding, whether they're non-coding, whether they're regulatory, and it's seen that above approximately 60% were non-coding, majorly belonging to the regulatory elements, enhancers and promoters and all of that, while 14 for PRS-CSX across other tools. So we also sought to find out the pathway enrichment using Reactome. So this is for bridge PRS. Found that it was the only tool that brought out significant enrichment, and this was an interesting finding because it shows that these genes being enriched are actually biomarkers used in prostate cancer screening tests, and the flow that's the prostate cancer signaling axis, and this have been verified from literature as well. Now to our nice web application. I think I'm being biased. So the web app, this is the front page of the web app currently hosted on SiteGen Africa. So the user goes to the web app, select PRS tools from a catalog, like shopping, depending on whichever you want to use, and then live run. It shows the live run status and the timeline. When you pre-process, you process PRS execution and all of that. And then it also shows, after evaluation, shows you plots. And then we also ran a user satisfaction survey and found that above 80% of users were satisfied with the PRS. So our main vision for Transcest PRS is that we also want to extend it to include quantitative traits, because right now we only tested on binary traits. We also want to test on psychiatric traits as well. And also we want to include more multi-ancestry tools, majorly tailored to add mixed populations, because the tools we have currently are tailored for multi-ancestry. There are some tools that have been built like Gaudi, Howdy, and all of that for increasing prediction for add mixed populations. And those ones, they target more refined linkage disequilibrium panels. And also one major goal for Transcest PRS is for it to be adopted in capacity building initiatives. So for users to be able to carry out polygenic risk score analysis easier, and for them to learn via our web app. Thank you so much.
Thank you very much. Can I call all the speakers up from that session, including you, for your questions? Where are our speakers? There we go. Okay, so the floor is now open for questions to any of the speakers from this session. Okay, while you're all thinking, I have one question for Alex. Those graphs that you showed with a huge increase, so how much of that is the same proteins, more granular, as in more different domains, versus actually increasing the protein that, you said it was 78% or something. How much of that actually covers more proteins that were never covered before?
Do I press the button to have this switched on?
It's on.
Okay. Yeah, great question. Actually, I don't know the answer. We're doing a mix of both sort of gap filling new domains and building new ones. So I think the answer to that one is I'll ask Claude to write a script and I'll let you know later.
Okay, thank you. Questions from the audience? We've got one over there. You could just say which speaker it's to.
The first one.
Okay, Alex.
Yes. Why is it necessary to develop more protein families or PFAMs?
Yeah, why do we need all these families? So essentially, they provide a way to sort of organize our knowledge about proteins, and sort of knowing which ones are related to each other can help you understand what their functions might be. So you might have a protein that you've discovered in an experiment is a hit, but nothing is known about it, except if you go and look in PFAM, you find that it's related to, kinase receptors. And so then maybe you understand that it's involved in signaling. We also have lots of sort of proteins families where we know nothing about any of the proteins in the family. We call these domains of unknown function. And so those have been quite a target for experimentalists and structural biologists to go and try and discover what the function is. So there's sort of lots of different ways this information is useful.
Okay, so we can take more questions. Let's start from here.
Yeah, so thank you very much, Shona, for the excellent talk. My question is about polygenic risk scores. And as we know, we have a huge genetic diversity in Africa. Should we think sort of different PRS scores for each ethnic group in Africa?
Can you repeat the question? Sorry.
About the polygenic risk score. So we have a huge genetic diversity in Africa, right? So should we think about developing different polygenic risk scores for each group or ethnic group in Africa regarding the diversity that we have and the differences between the sub-ethnic groups in Africa?
Okay, so let me repeat the question to be sure that I get you. So you're asking if we should gather the different ethnic groups within Africa before doing polygenic risk scores?
So what she's asking is that would you have one polygenic risk score for the whole continent?
Exactly.
Or would you need to refine it for every region?
Yes. You need to refine polygenic risk score for each region. So that's and that's one problem we're facing in polygenic risk score, and that's what we call transferability. So sometimes when a polygenic risk score is trained in a population where most of the data was trained from European, maybe largely about maybe 70% or 80%, it's going to lose its predictive accuracy or power when it's applied to a different population from that. So that's one thing we are doing in polygenic risk scores. So during the training, we try to make the data set more diverse, including we want to include the populations where it's going to be tested on, because that would increase the accuracy better. And then there are other ways actually using finer or better linkage disequilibrium panels. And yeah.
Mm-hmm. Okay, thank you.
Hi. Oh, this is working. Mugume, excellent presentation. My question to you, I love the usage of self-hosted models. Have you done any benchmarking for different self-hosting models? Or in general, what's the strongest self-hosted model that you've found that works best in your workflow?
Thank you for that. So yes, we have tried a couple of models. So generally, we found that for some use cases, depending on how complicated your data is, the smaller models, the quantized models, normally have a problem with interpreting the instructions. So based on the infrastructure you have, we found that bigger models, let's say like maybe 27 billion parameter models, they worked better on some of these, tasks, because the task that we are trying to assess is not that complicated for those kinds of models. So yeah, so if you're really thinking about deploying this application, you'd think around those kinds of parameters. Yeah.
All right, you can go ahead, sir.
Yeah, thank you very much for your excellent presentation. So I have one question for Alex. So I used to use PFAM for domain, especially for predicting protein-protein interaction between human and also human and pathogen. And I remember that we faced some problem to access to the data. Now, when you're using TED, so how to access to the data? Is it available freely on the website or yeah?
So yeah, with PFAM, perhaps you're referring to, we switched off the PFAM website in the past. And we did that because EBI was running two different protein family databases, PFAM and Interpro. And that seemed slightly crazy from one team to be hosting two different websites. So we moved all of the hosting of PFAM data into Interpro and sort of tried to make the Interpro website do all the functions that the PFAM website used to do. So yeah, that's sort of a change. So PFAM is still running. We're alive and kicking, and the data is all freely available through the Interpro API. And the same for TED. Actually, it has an amazing API. So we're sort of just gathering the data straight from there. So yeah, it's all there and available. Happy to answer any other questions later.
Thank you. I have a very quick comment and a question. Ms. Osalu, amazing work. I look forward to using this for my personalized medicine certificate students to learn how to utilize. Thank you for that resource. And Dr. Zdrazil, I have a question, is more of a, collaboration. How do you engineer the socio-technical aspects of your community? I'm impressed to see how you have managed to standardize the capture of metadata for your resource. And I wonder how challenging it has been to get your community to adopt these standards, or what has been your strategy to making sure that the community for Kimball actually adheres to these?
Thanks for the question. It's a difficult one. So I'm not sure the community always adheres to standards, right? So I think Campbell just sets an example because it's a database with a very long history. It's the main database used in drug discovery research in Europe. I don't think there is any competitor like this. So this is why we kind of set standards, but if we really could also convince the community to follow these standards, I'm not sure. So, but certainly one thing we want to do is, because there have been several papers being published in the past 2 years or so, where people that build machine learning models, they would always, of course, criticize, if, for instance, imagine a compound has been measured on the same target in multiple assays, people would say, oh, why do we see different bioactivity values, But of course, there's something like inter-lab variability, and even if the same person repeats experiments, they will. So, there's some variability. So, and then people publish about this and say, oh, the data is maybe not so high quality. So what we are thinking of is publishing curation guidelines for the community, because there's curation we do, but then there is also some pre-processing or curation that the user wants to do when they establish their data set, right? So we want to now really be a bit more proactive to guide users more how to best make use of our data, so like best practices. I don't think we have done this so much in the past because there's so many ways you can do it. So we left people the freedom to do that. And they still can, but we can provide some more guidelines. So I think that's how we want to go forward. I hope that even answers your question.
Hi, thank you, Nikki. I have just a quick question for Miss Blessing. Thank you for the very nice presentation, really good work. Maybe I missed it during your presentation, but did you test the trans PRS on different populations, and for which disease?
Yes, it was tested on prostate cancer and population, African population, just African population. So it was built with multi-ancestry data from Europeans and Africans that was used to train the model we predicted on African population.
So you took GWAS's data from European populations?
And African population.
Right, because there are, recent data on prostate cancer in Africa that have been published recently. Did you include those? No. Did you include any other data on prostate cancer from Africa?
Yes, so let me explain it again. For the GWAS data, we got our discovery data from GWAS catalog, so European data and African data. So one good thing about these multi-ancestry tools is that it leverages the statistical power from the European population and then uses it with the African population discovery data. Then we tested on African populations. We've not tested across, East Asians and Hispanics and other populations, just Africans, and that's part of our future work. Thank you.
Thank you. Thank you so much. So great talks, all our speakers. So my question is mainly for Alex and for all the speakers. So despite the boom of the AI and the usage of the AI, we saw it from your graphs. So we will always starting point would be a human and the ending point would be a human. And as you mentioned, I mean, maybe the creators, what other skills will be mandatory for future curators and researchers? And you mentioned that maybe the researchers will be a software engineer also too, so it will be some kind of thread of develop for the developer. So what will be the skills to master or maybe to have the best of this development of the AI?
Yeah, great question. I mean, you mentioned that the human is the starting point and the human is the ending point. I've learned with AI never to say never to anything, because as soon as you say, oh, this can't be done or this is challenging, then a few months later, it seems to be normal. So, but the skills of by curator, I don't think really change. You know, one of the key ones is being critical, so not unplugging your brain. And so in the Frederica's presentation about Amos, this obsession by detail and being able to spot errors rapidly. These are all skills, but this just converts into an AI-based process. Actually, when using AI, it can be incredibly difficult to track down how it got some piece of information. Where did it come from? Because they can be quite like a black box. Some of the reasoning models, so they do all this thought, but the frontier models don't actually let you see what that thought process was. So, I think we've heard from some of the other presentations, still the use of dictionary approaches, and these things are explainable and you can understand what's going on. So I don't think we should completely sort of bow down to our new AI overlords and, try to keep it all understandable as far as possible. So yeah, the skills haven't changed, but the tools that we've got available, I think, have improved a lot.
Okay, so we've got a question over there, and then we'll have probably one more question here.
Can I go first, or somebody came first? I think I'll allow the student to go first.
I also have a question for Alex. You mentioned something earlier that proteins predict molecule structures accurately, and that structure prediction makes protein classification easy. So my question is that, can these prediction help create new medicine, or are they mainly for research?
Sorry, I missed the very last bit of the question. Could you repeat that?
The question is, can these prediction help create new medicine, or are they mainly for research?
And create new medicines.
Okay. Yeah, that's, I suppose I'm focused on this very research sort of molecular biology part, but can you use this to do medicine? So I think the answer is yes, these tools are beginning to be useful. So the sort of the development of drugs is this, decade-long pipeline of different steps where you try something out or you screen lots and lots of different drugs, and lots of those drugs fail as it goes through the process. And it costs billions of pounds to develop these things. And so, MBI, we have this project called Open Targets, which is sort of trying to is designed to be better at working out which drugs might actually work and cutting down the time. And so lots of people are trying to use these AI methods to, work out what drugs might work, which drugs might be actually harmful, and so you shouldn't try taking them through clinical trials. So yeah, it's a big whole field, and probably Barbara's much better placed to talk about this than I. Yeah, I'm not an expert.
No, thank you very much.
Yeah.
Just to thank Blessing for a fantastic piece of work. And I think it underlines, obviously, as everybody can see, the key challenge we have in Africa. You mentioned, obviously, the Eurocentricity of the models, and obviously the issue of fewer data sets, unavailability of large-scale genomic data sets centric to Africa. Those are obvious and clear. Would you maybe want to say one or two more things that your work has uncovered in terms of the challenges in this specific work, and any possible policy recommendations or any other recommendations that you think can unlock the space? Thank you.
Okay, thank you so much for that question. Like you rightfully said, there are actually so many challenges, concerning the adoption of polygenic risk scores in the clinic, and one of them is the equity gap. So the dataset angle is one of the most talked about angle, and that's the angle that most, should I say, many researchers, wet lab researchers, focus on, because we need to generate more data. So my angle for my research is the methodological gap. So my supervisors and I, crafted this work focusing on the methodological angle, because we found that there are actually tools that target those problems, the multi-ancestry tools, and as I previously mentioned, they leverage the statistical power. We're not saying that just because they are European data sets, they are not useful. Actually, the reverse is the case. It's actually very useful, but the multi-ancestry tools leverage this. But another thing the multi-ancestry tools do is that they also include linkage disequilibrium panels for each of the populations that the user is interested in, and we have that integrated in Transcend PRS. So depending on the population you are trying to benchmark or calculate PRS for, you can choose the specific LD reference panel. So currently we are using the 1000 genome LD reference panel. There's also the UK Biobank own, but we are using for 1000 genome. So you can actually choose the LD reference panel. And one reason for doing this is that we all know that, or let me not assume that, the linkage disequilibrium patterns among Europeans and Africans are actually different. For Africans, we have shorter LD reference panels. For Europeans, they have, higher LD reference panels, or longer LD reference panels. So same thing goes for allele frequency sizes. Genetic architectural patterns, they differ largely. So these are some gaps or patterns observed, but from the methodological angle, we decided that let's look at those tools and how they solve these problems, and then let's benchmark, because benchmark is one of the key determinant factors that determines whether a score can be trusted. So that's why we're benchmarking. So yeah.
Okay, so we have 20 seconds we have a quick question from the panel, and a last question, and then.
Yes, a quick question to Dr. Bateman, please. So it's regarding the Claude, that you are using. So this is, I suppose, a public one. When you are uploading the papers there, so are they open access papers? Because okay, so you're not uploading those that are not open access.
Yeah, all the work that we do is with the open access portion. Well, actually, so when no, I'm not sure that's entirely true. So I will upload PDFs that I may have access to. if I'm just doing a sort of specific
But
prompt.
will that be, for the LLM, they are going to keep it? So is it something that we can do at our?
So yeah, that's a great question. I don't know the specifics of that. I think most researchers around the world will be happily uploading PDFs and not thinking about the IP issues. So, I mean, Anthropic have got into hot water legal trouble because they stole the copyrighted material from everywhere, basically. So I suspect there's nothing that I'm uploading that they don't already have. But yeah, that's not really the right excuse.
All right, I will have to take the last question, and my question goes to Dr. Anissa. While going through your biography, I realized how much you've contributed to the scientific community. So I would just like to ask more like a career path question. So what is that one soft skill that you would recommend to younger generation coming up in that path? Thank you.
What is that?
That one soft skill that you would actually recommend, to the younger generation coming up in that career path. Thank you.
Skill. Skills?
Yes, one soft skill you would recommend. I mean,
Skill.
yes.
To the, yeah, okay. I mean, coming from an informatics background, so I think any discipline, whichever discipline that it is, whether it is science, whether it is, for example, law, whatever it is, even finance. So one has to get the IT skills and AI skills, even if it's not part of the curriculum. This is something that you can go and learn on your own, because you have to have these skills. Without this, any discipline, it doesn't matter which discipline. Of course, we informatics, we need to be at the forefront, of course, but not sure if we are able to keep up. But yeah, that's the skill.
Right, thank you, ma'am. So we've come to the end of the session. Let's kindly appreciate all the presenters, please. Let's appreciate the presenters for their time and their efforts. And we also appreciate the audience for taking your time, for being attentive and for making this as engaging as it is. Thank you, everyone.
So the panel just needs to stay here for a photograph. Any other announcements?
So we will have lunch in Ablu in the reception hall, but we're also trying to take a group picture before we get lunch. So just go, it'll be at the pool where lunch is. So we'll ask everyone to try and move towards the lunch menu as quick as possible.
Sorry, before you go out for lunch, hi, my name is Kutala. I just want to do an announcement for mentors. Thank you for volunteering to be a mentor to our next generation scientists. Can I please ask you to sit at the table at the back? I did send a list of learners that you are assigned to and the table that you are assigned to. Please go to the back of the restaurant. You'll see table numbers on top of the table, please. Thank you so much. Enjoy your lunch.
Welcome back from lunch. I hope, oh, that's okay. I don't mind. I'm always short. I hope you all enjoyed the nice meal and a little bit of outside time and some sunshine. We're going to get going again with our session 2, methods and techniques in biocuration. I'm one of the co-chairs. I'm Lynn Shermill. I run the Human Disease Ontology Project, and my co-chair.
I'm Tim Tokwe from Nigeria, and I'm also a bioinformatics postgraduate student.
These are short talks, 10 minutes, so we're going to very brief introductions. Rachel Lynn's going to come on up from Cosmic, and we're going to hear about your resource. And then we have 4 speakers in total, and then a panel session.
Okay, great, thanks very much. I hope you're all awake after lunch. So I'm going to talk to you about how we've applied an AI pipeline to identify curatable papers in Cosmic. So the Cosmic Knowledge Base, also known as the Catalogue of Somatic Mutations in Cancer, it's an expert curated resource for exploring the impact of somatic mutations in human cancers. So I'm going to focus on our core curation, which is where we identify somatic mutation data in publications. So we identify the publications, extract the data and standardize it by mapping to internal controlled vocabularies, external ontologies, and then present that on our website and also provide very large downloadable data files. So my talk's going to focus on how we identify those curatable papers. So we have quite specific requirements on what makes a paper curatable. It must have mutation data, somatic mutation data at the sample level. Those mutations need to be mapped onto a gene or directly onto the genome, depending on the type of study. And of course, it must have a detailed cancer classification of histology, preferably with the site of the cancer as well. On top of that, we try to record as much metadata as well. So for example, at the patient level, we may be able to record age, gender, ethnicity. At the tumor level, perhaps the stage and the grade and the drug response. So we try and record as much data as we can. So we have 2 sort of streams of PubMed searches to find these papers. We have weekly searches where we search specifically for 272 cancer genes. We have been using PubCrawler to manage these searches. This can return us about 200 to 400 papers a week. Then for each sort of biannual release of Cosmic, we have a specific disease focus where we'll search back for several years. And this can return us several thousand papers. All of these we need manual review to determine whether they have the sort of data that we want to curate, which of course can take several hours per curator per week. So when the opportunity came up for an AI hackathon at the Sanger Institute, where we're based, we thought this would be a really good area to try and focus our efforts. This was partly also determined the fact we had a ready-made training set. We had a corpus of over 21,000 papers collected over the 20 years that Cosmic has been around, which were classified as curated or had been rejected due to the relevant data being missing. So our software developers went off to this AI hackathon. They assessed 4 models from Hugging Face, PubMed, BioBert, Mistral, and Llama. These models were trained using the title and abstract of these training set publications. So I won't go into detail on each of the models, but they decided that the PubMed BERT model was providing the best predictions for whether papers were curatable or should be rejected. So they incorporated this into a automated pipeline for us. So now we have an API that runs our weekly cancer gene searches, passes these through the AI classifier that they created, and provides the curators with a report each week. We also have direct access to the API for running our more ad hoc disease-focused searches. So every week for the specific cancer gene searches, the curators receive a report which details the queries that they are in charge of, the publications returned, and then a status for each of those papers, whether it's curatable, can be rejected, but also a score. So if there's any edge cases, they can be looked at a bit more closely. So over the next few months after the hackathon, we assessed this pipeline more closely. We collected data on the true positives, false negatives, false positives, and true negatives. And for the weekly searches, we found that the model performed really very well, sort of 85% precision. So the number of correct predictions and a recall of about 94%. When we looked more closely at the ad hoc searches for the disease focuses, so in these cases, we often run queries sort of more specifically to try and find whole genome, whole exome studies, as opposed to targeted gene studies or case studies. Now, in these cases, they're obviously larger data sets. The data tends to be in Excel files and supplementary data. And what we found for these studies was that the model really wasn't performing very well at all. Most of those papers that came back classified as curatable were actually not curatable. It was over 50% false positive rate. Now, looking more closely, this wasn't really the fault of the AI classifier. It was that age-old problem in curation that we discussed in some detail in one of the workshops yesterday, but the data not actually there. It might be just an oncoplot. The raw data may have been just put into a repository as BAM files, which we don't have the capacity to process. So, but this did highlight to us that, one, we need to keep pressing people to provide us with the data in the correct format, but also that we should train our model more specifically for these different types of searches that we need to do. So in these cases, we need to include the full text and supplementary in our training data. Just to give you an idea of just how much this AI classifier has improved the efficiency of our curation workflow. So this data is from January to November of 2025. Our weekly gene searches returned nearly 13,500 papers. Using the AI classifier, that reduced that to just under 3,000 papers that we needed to look at. We don't, unfortunately, have capacity still to curate 3,000 papers. So we still need to do some further triage on this to identify those papers that will provide the most value to Cosmic. But by having this much smaller set of curatable papers, we can now sort of look more closely at the curatable landscape, the cancer landscape. We can use existing tools such as Europe PMC, PubData. So, for example, PubData provides AI-generated disease annotations to publications, map to mesh, so we can use that to filter these papers further using the mesh hierarchy, the cancer part of the mesh tree, and visualize this data. We can also map that back to Cosmic via NTI terms. That's not perfect, but we can work further on that to identify papers which have histologies with low representation in Cosmic. So now we have some nice tools, a little Streamlit app at the moment, which just allows us to view the histologies being returned within this curatable set. So we can more easily look at the landscape, for instance, over a year, and then home in on histologies, papers with histologies that would actually want to curate. So, for example, we can quickly see that in February last year, there was a paper for male breast cancer, which is one we'd want to prioritize. So this AI pre-screening has saved us many hours of work, allowing us to focus on our key curation tasks, and has the added benefit of allowing us to monitor the curatable landscape of cancer data and to set priorities for our curation. We will try and further refine the model through feedback retraining to reduce the number of false positives and negatives, and also to address more specific use cases, such as when data is missing in the whole genome and whole exome screens. And of course, this historical saving of both positive and data did give us a massive advantage here. We had a ready-to-go training set. So I feel like we've just sort of touched on our applications of AI within our curation workflow. We've already touched on identification of curatable papers. We are, of course, looking at the whole curation workflow, where else we can apply AI. And we're also looking at other parts of Cosmic. So we also have an actionability project, which looks at drugs specifically for somatic mutations in cancer. So in this case, we're not looking at publications, we're looking at clinical trials, say, for example, from the clinicaltrials.gov website. So this is a slightly harder problem because we don't have the pre-trained the domain-specific models like PubMed-BERT. So our software developer here is having to develop the LMM firstly to identify whether a trial is actually a cancer trial, and then create a very specific customized prompts with a lot of very specific rules for each biomarker to determine whether that biomarker is actually within that trial, within the context that we'd want it for curation. So I don't have any performance metrics yet for this data, but the actionability curators are getting reports now so that they'll be able to assess how this, this classification is working. Okay, so I've actually finished in time. So thank you, sorry. I should just say thank you to the rest of the Cosmic team. Those in bold are the ones that have been more involved in the AI project. And a particular thank you to Sumod and Andrea, who actually probably did the bulk of this work. Thank you very much.
Thank you very much, Dr. Rachel, for that wonderful presentation. Our next speaker is Professor Yasma Vakim. Professor Yasmina Vakim from Mauritius. Thank you. Let's welcome her.
Right. Good afternoon, everyone. Thank you for the introduction. I am Yasmina Fakim from the University of Mauritius, formerly from the University of Mauritius, now working on AfriGen-D project. So I'm here on behalf of all these members here of the reference graph working group of AfriGen-D project. So basically, I will just quickly explain to you what is the aim of this project. We all have been doing variant calling, finding mutations, variants in genomes or in specific regions of genomes, and using what is now very popular, which is the linear reference which has been available for some time from the Genome Reference Consortium, and which has been updated regularly because of the missing parts and gaps in that reference. But this HG38 has been very useful and is still being used widely. And now we also have the T2T telomere to telomere reference, which is from the Pangenome Reference Human Pangenome Reference Consortium. Okay, so basically those resources are here because we need to align new genomes, new sequences to a reference in order to identify mutations and variants and therefore assess diversity within genomes. And the theme of this meeting here is from data to discovery. Obviously, when we want to curate, we want to look at a specific sequence, we need to have accurate data to do so. So with the linear reference, there is a lot of shortcomings because it can only represent one letter, one allele at a given site. And obviously, when we align diverse genomes, which are not very close to the reference, the linear reference, which we know is coming from 65% of the GRH, 38 is from one individual with bits and pieces from other individuals. So it doesn't actually represent even one population, not even the Caucasian population. So we use them for a read alignment and therefore variant calling. Just quickly, I mean, what is the aim? The main aim of this project is to produce a reference in a form that would be better adapted for variant calling from populations that have very high genetic diversity. And here we're working on African genomes, which you all know are the most diverse in the world. So in order to have a better and improved representation of different alleles at a given loci, the approach is to use a graph representation. So here, by having a graph representation, would be able to compare a new genome or a new sequence with multiple genomes from many individuals. So this representation would allow us to have an approach that have the representation of many alleles at one given locus. So hopefully that's what we'll be able to do. Quickly, for maybe some of you are familiar with graph representation, maybe not everybody. Graph representation means that we can connect components, single components, where every pair of vertices have a path between them. For example, here, this is a simple representation. A is connected to C, D, E, and whatever. So what is meant by a pangenome reference graph? So this comprises of the two terminologies, pangenome. I mean, a pangenome, as we know, has been around for some time, trying to get, all the sequences of individuals of one species together, because we know that all individuals of one species are not 100% exactly the same. So we want to represent everything that is in that one species. And in doing so, therefore, we have a pangenome. Now we have to put it in a form that can be used as a reference. So this is where the graph representation is sequence graph, which is the element. And then it has, of course, an index or a position and their relationship and connectiveness. So I know this doesn't maybe mean very much to you if you're not familiar, but once we explain this in terms of sequences, so here, in a linear representation here, we find, let's say these are 3 sequences from 3 individuals from the same region. And as you can see, the colored parts, they cannot all be represented in one sequence, one linear sequence. Okay, so we can look at them in this form of a matrix here, where if we take this little bit of red here, we see in the first genome it's here, in the second genome it has moved further down, and in the third genome it's missing altogether, right? So, and this is the representation of those sequences in the form of a sequence graph, okay? So the point here with the graph is that the common sequences that are exactly the same among these 3 have been collapsed. They have been collapsed into what is known as in, sorry, this should be a node, this is an edge. So the sequence graphs improves the representation of diverse genomes. So the bubbles is where the diversity is, where they are not exactly the same. So this is known as a bubble or a snarl in graph terminology. And then the other terms that I use are graph length, path, and so on. And just for the sake of time, I'll move a bit quickly so that I can show you some results that will illustrate what is explained here. Yeah, so this is again a representation. The snarl or the bubble is here, and the edge. So the edge is wherever the sequence are exactly the same. Then it would be called an edge. Now, the nice thing about graphs is we can, from the graph itself, we can immediately identify haplotypes, right? So individual haplotypes would be, the information on the haplotypes would be available from the graph, okay? So there are various tools that have become, that have been developed to do so. I'm not going to go into all the details, but it is one of the 2 most currently used tools for building the graph. So what do we mean by building the graph? So let's say we want an African reference graph. We'll need genomes from different populations in Africa. And then we take these genomes and we build the graph. So these are the tools that are used. So building the graph means aligning them all together, collapsing all the regions or the bits and pieces that are exactly the same. And wherever there is diversity, where they are not the same, then they are in a bubble, right? They are represented in a bubble. So this is one of the tools. And this is another tool. These are the 2 most commonly used tools, and their approach not exactly the same. The MC graph, the mini graph cactus tool, actually is, it will remove all the complex regions, all the centromeres, telomeres, segmental duplication, and so on. It will remove those very complex repetitive regions and use the rest. Whereas the previous one, it will force all the genomes to come together and will force them to align and produce the graph. So it might not be very, realistic in some regions. So our project, actually, we build a few graphs using African genomes, our own African genomes, and those are from the HPRC consortium and these others. Okay. So what have we done? We've got some graphs, MC graphs, PGGB graphs. So in here, you can see this is for each chromosome. Sorry. For each chromosome, you get the number of nodes, the number of edges, and we can compare among each graph. Okay. So, and we have also means to assess the quality of the graph, how good it is to represent these genomes. And some of the work we have done is extracting regions of the graph, so we can actually extract regions of the graph and make smaller graphs and look at them and explore them. And this is what we did for some of these genes, the highly polymorphic genes. And we see again the lengths, the nodes, and so on. This is another way of visualizing the alignment, the graph alignment, with the various, the different positions where there are variants. So you can really see some of the structural variants. So basically, graphs are, performing much better than, of course, linear reference. But the challenge now is to get them built in an appropriate way. And you can see here, these are different haplotypes. Okay, in one visualization, we can see different haplotypes. Okay, we did some work on structural variation, on variant calling. So just 2 seconds for me just to highlight this one, because this is where we see really how the graph outperforms the linear. So in orange, here is the linear, and this is the MHC, the region. Okay, so basically, I think I've covered everything. Thank you.
Thank you so much, Professor Yasmina. Our next presenter is Professor Bastian from the Arctic University. Please, let's give a round of applause for him.
Yeah, thank you very much. This is quite a mouthful, so I'm essentially presenting our work in the last 10 years on manual microRNA annotation and curation in the database MuGeneDB. And just to give a little bit of a disclaimer, I'm very happy to be here. It's my first Biocuration meeting, and it's not going to be the last one. And I took some traveling, but it was all worth it. And I guess now even the sun is coming out. That's even better. So very brief introduction. We heard some intro already today. MicroRNAs are short non-coding gene regulators discovered in 1993, awarded Nobel Prize for the discovery 2024. And what they're doing is they are post-transcriptional regulators of messenger RNA translation. So, and very important regulators of development. But in 2001, so 8 years after the initial discovery, it was discovered that they are not only restricted to C. elegans, but actually deeply conserved in many animals and also in plants. Our work focuses for now on animals, and which kind of kick-started a real explosion of the field in 2005 was the discovery that there's a substantial downregulation of microRNAs in many diseases, including cancer. Now, as for today, we know a lot about microRNAs. There's about 150,000 papers describing their function, describing the microRNAs in different organisms. And we are quite aware of their biogenesis. So what is the problem then? The problem is that the criteria for the annotation of microRNAs were defined in 2003. and that is pre-NGS. So the amount of data that actually came after this kind of overwhelmed the system. And what we did and the vast majority of these papers that you see here are actually from the disease field, so from non-experts, no offense, but not microRNA experts, but people that are interested in the role of biomarkers or disease detection. So I work at the Natural History Museum in Northern Norway, and I did so throughout my career because I'm a zoologist. And just as Natural History Museum collections, gene repositories and databases require curation. I don't need to tell you that, but this is a standard slide that I have in my presentation. So people get the idea that when someone comes by at the counter in our museum and has a nice rock and says like, I found this fossil of this shell here, please put it in your collection. Our mollusk curator gets it and say, thank you very much for this nice rock. It indeed looks like a mollusk, but it's not a mollusk. Right, so this is what we did. We looked at a couple of problems with the previous microRNA repositories out there. Here, MirBase, we have idea that we now understand the evolution of particular microRNAs quite well, but we see that, for instance, the same gene has many different names in different organisms. Even within the same organisms, onologues have different names. We have collected the biogenesis criteria into a framework, bioinformatics framework, and tested whether or not these criteria for their biogenesis, which are quite strict, are fulfilled. And we found that about a third of the microRNAs described in human actually fulfill those. So what else is that I told you that in 2001, kind of the back-to-back science paper really said they're deeply conserved and they are used as phylogenetic markers. So now this is quite surprising when you look at very closely related organisms that they should have many different, very different microRNA complements. So, but of course, repository MirBase had one problem, and this is that the field grows so fast that obviously, I mean, I spoke with them a lot, and this is not an attack on the work, but it's impossible to deal with these submissions. So there is thousands and thousands of microRNAs described, which mostly are not microRNAs. And people, including us, have called this out and tried to warn the field, especially the biomedical field, to not use those annotations for biomarker discovery or for disease understanding because they are not going to help them. And in 2015, after having been my opponent for my PhD, I teamed up with Kevin Peterson from Dartmouth and we re-annotated the human microRNA complement. Annotation here is, of course, not functional annotation, just is it a microRNA or not? What is the precursor? What is the mature sequence? What is the star? And we also tackled, and that was the biggest amount of work, the naming issue, because if genes are not named the same way within the same organisms or between two organisms, you have no way to understand their evolution or their functional roles. We had since then, and we founded MirGeneDB, the manually created microRNA gene database. And since then, we had a couple of new releases, and we are now at 114 species and 25,000 microRNAs. The purpose of the whole endeavor here is not intrinsic curating, but we want to understand animal evolution and the role of microRNAs, so that our goal is to have at least a couple of representatives for each animal phylum in our database. Right, and you can do beautiful things, and you can see that actually microRNAs are really super strongly conserved and have patterns that are specific to particular animal groups. Okay, so now we know this. Let's help people to kind of find microRNAs, study them in their organism of interest or in their disease of interest. And we tried this by answering particularly important contributions for biomarker field and wrote these papers that to kind of warn people that what has been published might not be real. And you can see here in this case where we had a correspondence in the New England Journal of Medicine where the authors described this to be a very important microRNA involved in myocarditis. And you don't need to be a microRNA expert to already know that this is not a microRNA. This is an artifact of their analysis. But to be more constructive, we then now formulated a couple or kind of a white paper, I hope. I hope the microRNA field kind of catches up on this. We're sending this to a lot of journals, to editors that we know that they could simply check. And I'm not talking about curating what authors claim to be right, but to simply check on a couple of things. Annotation, the targets, genome representation, and also expression, because you'd be surprised that many microRNAs are actually not even found in a genome. Okay, so we have this checklist, and I'm not going to go through all the details, but please, if you are interested, find the paper or approach me and we can talk about it. We show these examples of good and bad guys, and what's more is, of course, we want to use this because 114 species in 10 years is great, but there's now, I just updated this yesterday, there's more than 12,000 animal genomes out there now, assemblies. So you can do the maths yourself. That would take us decades, if not longer. So we have a paper recently published called Mir Machine, a tool where we develop based on our manually created microRNA family covariance models, and we show that these are working without small RNA-seq data to predict conserved microRNA complements, no novel ones though. And we are now following up on this, and I'm sorry, this is a bit blurry. This is work of my PhD student, Vanessa. She has, in 1 week, annotated 760 genomes for the microRNAs. So it's a bit like what Alex said, time is running. If you're interested in microRNA annotation, there's this course coming up. And with this, thank you very much for your attention, and thanks for the invitation.
Thank you so much for that presentation, Professor Bastian. Our last presenter for this session is Mr. Takim Ezekiel. Mr. Takim Ezekiel, please join me as I welcome him. Thank you.
Okay, thank you very much, and precisely for the acceptation and the provision of the opportunity for me to come here and share with you my research. So I'm Ezekiel Takam, a researcher from the University of Geneva, and I'm precisely based at the Faculty of Theology of the University of Geneva. Yes, you got that right. I'm from the Faculty of Theology, and usually when I present myself as coming from the Faculty of Theology, I receive a myriad of questions related to my journey that developed a link between the theological background and then the biocuration / AI ethics / AI safety journey. And to have your answer to this question, I invite you to take part to the closing panel, which will take place on Friday. So I'm in front of you today, not to talk about God, but precisely about synthetic data. Or should I precise, synthetic dynamic data. Perhaps we can start with some numbers. I will say well-documented numbers in the fields of biomedical. Roughly 86.5% of participants in the GWAS cohort are from European ancestry, and the African population sits at around 0.5%. So sometimes the number can slightly change from 0.5% to 2%, going from studies developed in 2023 to studies developed in 2025. And usually when we use this reference databases such as the GWAS in order to develop polygenic risk scores. Of course, these studies perform at 4.5 times less accurately when applied to African population. As I mentioned, these are well-documented numbers just to showcase how today in the field of biology, the field of genomics, etc., there is a diversity problem. So there's nothing new here. For me, what is quite interesting is that nowadays, based on this observation, there are different forms of methodologies emerging, trying to augment the underrepresented population in these kind of databases. And one of these methodologies are the synthetic genomic data. But what is quite interesting is this. Nowadays, with the proliferation of AI, the generation of synthetic content has took an extraordinary speed. But our curation infrastructure, such as the reference databases mentioned above, or other databases that we are going to see during the presentation, has no policies, no labels, no detection mechanism applied to synthetic content. And the resulting risk is the entering and the corrupting of these databases by synthetic content, while also corrupting everything related to allele frequencies, LD, decay, etc. So the idea of our work was precisely to explore how we can identify this form of synthetic genomic data and then develop mechanisms to tackle them in the process of analyzing data that are provided to reference databases. So exploring the literature review moving from 2019 to 2025, we identify three main gaps, right? The first gap is precisely what we can call the over-reliance on statistical fidelity. So nowadays in literature reviews or in works related to this synthetic data and the analysis, the flagging of these synthetic contents, these work are basing themselves on the analysis of the statistical fidelity. And they just rely on the statistical fidelity of genomic contents, of synthetic genomic contents. But when just focusing on these statistic fidelities, they also have this tendency of marginalizing the biological plausibility, but also the privacy preserving criteria, which is quite important when dealing with the synthetic data for the sharing of data purpose. The second gap is the provenance gap. So we have surveyed myriad of, precisely 7 reference databases, and none of them, as I mentioned previously, has labels, policies precisely dedicated to the evaluation of synthetic content, or should I precise, synthetic genomic content. And then the third gap is precisely the absence of framework to govern these synthetic databases. So we have developed a small experimentation in order to show why this gap is quite, I would say, important, first of all, to identify, and secondly, to tackle. So we use 2 AI models, 2 AI methodologies to generate synthetic content. The first one is quite a strong model, it's called the Generative Adversarial Network. And the second one is a simple allele frequency baseline. So we generate, using these 2 AI models, we generate synthetic contents, synthetic genomic contents, and then we have decided to pass these synthetic contents through the quality check. And we have noticed that when it comes to the statistical fidelity, both contents were able to succeed the test. And we can identify here something like 0.99% of accuracy. But when now we try to focus ourselves on the biological plausibility, what we can call also the privacy preserving as well, we can notice that it's just the contents generated by the most sophisticated AI models, the generative adversarial network, that were able to pass the test. So the idea here is to show that if you were a person defined as a gatekeeper of these databases, and then we decide to fact check the synthetic data provided to us, just basing ourselves on the statistical fidelity, we would have accepted both contents while neglecting and marginalizing the biological plausibility, but also the preserving criteria, which is quite important when dealing with synthetic content. So this is precisely what we are trying to propose, and it's what we can call the Biocuration synthetic genomic data standards. And based on the 3 gaps identified, we are also trying to propose 3 main fields. The first point, the first pillar, is the minimum validation. Here we just want to show that it's important for us not only to focus on statistical fidelity when evaluating synthetic content, but also take into consideration the biological plausibility as well as the privacy-preserving criteria. The second pillar is the provenance metadata. When we are a database and then we receive the provision of content or synthetic content, this synthetic content should be able to integrate in their description everything necessary for a curator to work. We are talking about the generator names or version used to generate the content, the training data used to train the models to generate the content, the validation summary, the watermark method or dictator if exists, and then the most important thing, the explicit epistemic status that flag the data as synthetic data. And then the third point, the third pillar of this standard is precisely the governance approach, the equity-aware governance approach. And it's important also here to focus ourselves on everything related to the community approach. We have proposed 5 points in order to highlight this governance approach. The first one is the community consultation. If you want to generate synthetic data of a community, we have to consult the community. The second one is to develop mechanism to also govern the community, to govern the synthetic data with the community. The third one, is the independent validation. The fourth one, the benefit sharing. And then the fifth one, which is the most important and political one, is the non-substitution audit. That means we are using the synthetic data not as an excuse to refuse the importance of also recruiting underrepresented population in order to participate in real-world collection of genomic data. Thank you for your attention.
Thank you so much for the presentation, Mr. Takam. Can all the presenters step forward for the panel session? All right. Does anybody have questions from the presentation? Okay.
Hello, everyone. I have a question for Bastien. I have a question that relates your work to climate change. So can better micro RNA annotation improve how we study biodiversity changes and species classification in the context of climate change?
Thank you for the question. This is not our focus, but I think it is well possible that we will be able to understand diversity and phylogeny of organisms better, because we think that microRNAs are very good phylogenetic markers. Yeah, probably expandable to what you want. Yeah.
Thank you.
Behind you, if you could. I'll just.
Thank you. My question is to Bastien. Wonderful talk. So there is a study, several of them, where microRNAs have been used to kind of characterize different cancer cell lines. So, in your opinion, can we, how faithfully can we extend this analysis if we look at single cell type in plants, where we can actually start to characterize different cell types using microRNA profiles? Do you think they would be able to, because cancer cell lines are different phenomena, but normal cell lines, normal cell types is a different thing. So any comment on that?
Yes. So where we are not working on plants, in animals, it's extremely well understood that sets of microRNAs are very specific to tissues, organs, or even cell types, not focusing on cancer cells here. And I think, as a matter of fact, that microRNAs contribute to the evolving and increasing complexity in many animals, such as mammals and so on, in their number of distinct cell types. So for plants, we know that if you take a leaf and you have the right hormones, a new plant will grow. You should not try that with an animal. So I think what is a cell type is not as narrowly defined in plants, but it could work. In animals, it works. It's not a cancer thing.
Yes. Sure, you can.
Yeah. So that's a very interesting point that you posted, because in our understanding, the functional plasticity of the plant cells is much higher than animal cells. Sure. And in that scenario, so I mean, that was my concern, whether we will be able to find that kind of an distinct distribution of microRNA genes, because you know what, I mean, yeah. So thank you.
Okay. Hi, I've got a question for the first presenter, I think Rachel. Sorry, I couldn't catch your whole talk, but I had a two-part question. So firstly, you said you manually curate a lot, you get weekly about 400 to 600 papers, I think you said, that was manually curated. That seemed like a huge amount. So I was just wondering, how does your team manage that? And maybe if you could just summarize how AI has facilitated that process, because I missed that part of your talk.
Yeah, so no, that's the number of papers that we are returned from our PubMed service. We can't curate that many. So yeah, the AI is really helping us to narrow down which papers can actually contain the data that's curatable. But again, we still don't have capacity to curate everything. So hopefully in the future, AI will be helping us further with that, with sort of more data extraction. And
okay, thank you. I also have a question to the last presenter. The tone I got from your presentation was more of condemnatory or accusatory on synthetic genome, but I understand it's a very beneficial component on synthetic biology. So I don't know what comment you'd have on it, on the beneficial aspect.
Sorry, I think he said your comment or your presentation is more of attacking synthetic biology or synthetic genome. That you have the positive side of it too.
Absolutely. So, thank you very much. First of all, perhaps I gave that tone, but I'm not attacking synthetic genomic data. For me, I think it's quite important. It's a new revolution that will certainly going to help advancing the biomedical research. But the only problem that I've observed is we have framed the epistemic contamination, right? The entering of synthetic genomic data, and then the reading of these synthetic genomic data as observational truth, while silently corrupting real data. But to avoid that, our curation infrastructure has to anticipate. So the idea is not to deny the importance of synthetic data, but to anticipate the integration of this form of synthetic data in future databases curation. And for that anticipation, we need to define a standard. And that is a standard that we're trying to propose. So I'm quite positive when it comes to synthetic genomic data. And I think that's a huge revolution.
Okay, thank you very much.
Thank you. I'm Monica Munoz Torres. It's also a question for Mr. Ezekiel. As I was listening, I'm very much encouraged by the things that you're saying and the gaps that you're identifying.
Yeah.
And I would like to ask for some help in identifying, and just one of the gaps that you were saying is that no framework covers synthetic data.
Yeah.
Specifically, I am one of the workstream co-leads for the Global Alliance for Genomics and Health on clinical and phenotypic data capture, but I also participate on the regulatory and ethics workstream.
Okay.
And so there's like the genetic discrimination toolkit, there's the newborn screening tool. Help me understand, how is the regulatory framework for synthetic data going to be different? What kinds of things need to happen in order for this to be correctly identified that is not already present in the frameworks that are available?
That's a great question. Thank you very much. So first of all, I think the actual ethical framework that we were able to identify in our literature review, they are providing a strong blocks to identify this kind of problem or to tackle this common problem. And we have just highlighted the fact that these frameworks, even when they are already dealing with the idea of AI in their declinations or their recent declinations, but the notion of synthetic data and the problem precisely highlighted by the synthetic data has not yet been identified. And that is not just, that is not just in the field of synthetic genomic data, but we are also facing the same issues in synthetic contents related to politics, synthetic contents related to media, synthetic content related to all form of activities, right? But as I mentioned, we have to be quite proactive in order to also integrate a flagging, or should I say a precise attention to this new form of synthetic contents applied to our field of biology, biomedical, etc. So it's just an attention, but we already have the standing and the basic block, which is quite useful and possibly translated in order to be also tackling this precise field of synthetic data. Yeah.
Thank you. Are you okay with the question? Okay.
Yes, my question is to Rachel. So if I remember correctly, PubMed BERT is only trained on PubMed literatures. And do you have any insight why the other models are not as good as PubMed BERT? Yeah, why, any insight about that?
I don't really actually, but I mean, I could go back to our software developers who looked at the models more closely and find that information. But I don't know actually.
Any last minute questions? Or we're going to go to tea break next, or off the time a little bit. Sorry if you couldn't hear me. Any last minute questions? Because then we're going to go into tea break next. If we're good, there's a lot of really good discussion. I want to thank the panelists. Thank you for the questions. Ask the panelists to stay up for a minute. We're going to take a photo, and tea break, I assume, is right down the hall again. Perfect. We have to stay here. Down here? Okay. Oh, that's tricky. Down there. Scooch across. Here we go.
You should be here too.
Yeah, we should be in there too. Yeah. Just gotta, we may have to scooch to your right a little bit. If they want to. One more. Great, thank you. Thank you. It's a really interesting session. It's fun. Thanks, everybody. Thank you. So you're welcome, Jay. No problem. Oh, good at that. Yeah, it's fun to eat together. Here's the screen of you know, I would really there's oh, thanks, yeah.
It's coming up. My shadow is shooting. Oh, your shadow. Yeah. Okay, thanks. Yeah, like I have a facility. I will tell you what the light based the light the light is the light makes life please tell my dark. Yeah, he's here with your the darker one. Hey. The light. Oh, you know, it's the light. I don't want it to think. Because if I was zoomed into the picture, yeah, it's so you can't do the next group, something
okay, that's their mission. Yeah, so it's actually about an hour.
Yeah.
And then there's such a problem that we need.
Yeah, it's last week. Last week. I've been seeing your blog. Until the light is there. That's why you're paying so much. You don't force it. It's not like you.
So you basically, you see, the positive and then yes, that's one of the things that, yes, I did choose that for my work. But this one, I had to just double stop. And I get how I worked out before. And you kind of perceive it's very much the thing. See where you start. I've got to know how many was, and now I've got some spare. So that's the spider. So it looks like yeah. When you cast you just those two threads go it's that way and those threads are coming that way. Okay, so then it must be so this is pinhole. You put pinhole, you just hold it on like this. So that's making but firstly, I mean you need some tension. Well, yes, what you use. That's why you use the pins. So, you don't have big vision affiliation. That is that. And so this one I have oh, you can't see it. But there's sort of things all see the corners. So you haven't got to do. No. You every time you cross it over, you cross it over and stick it in and cross it back, and then go to the next thread. So you're continually. Well, so I've taken them out in here. So you basically don't take it. You can't take. You take all the things down. That's what I finished. That's where I started. Started out there, worked down. And I'm just thinking of time. Then I do it. Start. So when I get to about 8, I'll start the. Yeah, usually or you. So here, you would do that down in there, but and it will give you threads that are there. Yes. Yeah, so those go into there and those go into there, and then do that section, and then you do that section, which will be so you're work. And there's no point starting this point. And that's my bit for a couple of years, and I've been carrying it around in my life since then with all these bobbins that time. Okay, so no, when I was at primary school, it's one of the teachers, right? Very sensitive. But they're into I know that I'm definitely never I think that's yeah, it needs to be. And if you make a mistake, the thing so this is one of the me to learn how to be honey plant from honey. So here, basically start and here and things. You always do the same thing. You always do that well over those things.
Okay, so from here.
You have this clicker here. Just
Okay.
advance your slides.
Okay. Great. Thank you very much.
In a way, the only place I've shown is here. So like this whole thing, so this one looks like all the pairs are in it, no groups. Yeah. Yes, that's exactly why I don't even have any money for kind of like the five parts that we expect here is photograph. It's done in the first clear of the five parts that will show you what we need to be have the well, that's why this actual part here, see. Yeah. Yeah, I didn't know how it could work. Just first, I just interesting to see. This is half stitch. So if you see that line, that hair stays on. But when it came here, this is what answer and so this to be S. And to be honest, we should be less than they think it is because why? And you have fun together, but it doesn't really thing. So don't see like this. And I think see the lights being graceful and you don't actually see like you spot it, it's kind of big up there and it's very hard to see those great colours of it. With the light, when your eye takes it, don't really see that versus that. You would just see the wind going towards the moon. Last second. These full roads and it depends what's thinking for you. So of course there's a good thing. Yeah, that's definitely good. Well, what do you like? Because this is what the three exits and yeah. 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 No, it's very, yeah. Yes, and that. This makes yeah. This is like those two yeah. And then oh yeah, oh yeah, but then look at that. It's right. It would be a bit fun. Yeah, you just have to think about it as that's why you come to work out. So it is a bit of a big take. That's why I said this. It's hard. It's hard to really. It's a big ad, but it's a oh, you're on forward. I've got those from here. Yes, look, yeah. This one, white channel, because the threads will almost usually we can down the next. And this one starts to this one is on the end on this main pattern. So Yes, so it's kind of, yeah. And then there's this other thing.
And which one?
The next one. Okay, so you're basically, you don't really do the left, that is. Picture, I would get from one, but got that.
Okay, so. They will keep doing it.
Well, they're basically, what great market specifically, and I know, but this, that, this one here, so.
So basically. So this square.
Yeah, those red lines are where, so they're, yes, that's, you can see, I know what. So I have, in my own ground, the blue from sky. But in half, maybe in half, and they go on, so that you don't know. So they're exactly right, so it's. So this one, I use this dark blue to here. Oh no, yeah, that's this is. There's a spot. That's the next one. In the top, one word that's going way across, and was acid. Very understand. It's not really way to do it. Yes. So, it goes there. It does still go there. It's probably going to go over to that. You think the president looks to go diagonally. So it always comes to see where there's no any problem. But it's visible. Okay. Should be good. Okay. It doesn't go with the sun. Thank you. It is. Yeah. You know, it's just champagne planes. I'm saying about. It's a good coin. Yes. So this is my London. That's two. There are two different miners around the world. Yes.
Actually, what you told me.
Well, I just the problem we use through the.
Yeah.
Okay, but show them. So you, this is. I think And then twist, I just make those two more. And then fold them using one. But really, that's why this is. I think then on the time actually first I'm feeling lots of that. But maybe it's I don't think it's funny, put it down when there's. Then I see it. There you go. It's okay. There are mostly men. I mean, women, but there are some men who have I shouldn't. I guess yes, yeah. Yes, that's right. I'm going to take one. That's my next one that I'm starting, and it's going to have 8 game comments. It's much bigger than that.
The Tanya is there. But there will also be someone at the back.
So we don't.
Yeah. Yeah. So there's two booklets for you guys.
Amazing. Updated.
Yes.
Yeah, it's 4 o'clock. Okay. Hi, everyone. We'd like to get started. My name is Manadia, and I'll be chairing this session called Global Diversity in Biocuration alongside Dr. Charlize Ricks. So today, we'll be going for just around an hour with 20 minutes of questions. And let's just get started if everyone could take their seats. Okay, good. So here we go. As biocuration becomes increasingly central to global research and public health, it's clear that how we structure, standardize, and share data is not just a technical challenge, it's also a cultural and equity issue. Today's talks will explore what it means to build data systems that are both globally interoperable and locally meaningful. And hopefully this session will push us to think about diversity, not just in the data itself, but in the frameworks and communities that build it. So our first speaker is Dr. Malek Chouch. He is a scientist at the Institute Pasteur of Tunisia, whose work focuses on advancing genomics, bioinformatics, and data infrastructure across Africa. He's been closely involved in a number of major collaborative initiatives, including H3A Bionet, where he has served as a node ambassador, as well as being closely involved in the African Population Ontology Project, which works to better represent the diversity of African populations through more standardized frameworks. Malek is also involved in several large-scale efforts, such as the African Genomics Data Hub and the Psychiatric Genomics Africa Consortium and the Africa Biogenome Project. Alongside this, he co-chairs a training working group within the African Bioinformatics Institute, supporting the development of bioinformatics and data curation skills across the continent.
Overall, his work is centered on strengthening data systems, collaboration, and capacity in genomic data science across Africa.
Thank you so much, Monadia. I didn't know I did all these things. So, so welcome everyone. I hope you get some refresh after this tea break, coffee break. So today it's my pleasure to present the African population ontology, which I'm leading with my colleague supervisor, former supervisor Professor Ali Abel Kahla. We are based in Tunisia, and this work is part of the AfriGenD project. So the African population ontology, or AFPO, came to solve a practical, let's say, a practical problem related to, let's say, population genetics and how to annotate population in a consistent, searchable, and free, usable way for genomics study. So I don't know if who's of the audience working with genetic population, but usually some people working on genetics population are facing some challenges related to inconsistent terminology. So people are mixing terms like race, ethnicity, ancestry, and population. So those ambiguities can lead to some difficult comparison between different studies. And the lack of standardization and a controlled vocabulary and standard definition leads to data silos. So this lead also to critical consequence. There is some bias for geo studies. The work is not reproducible, not interoperable, and we can miss opportunity to do some kind of meta-analysis. So let's give a real word problem related to people annotation. So for example, we've taken the Yoruba population, which is a population based in Nigeria, West Africa. So in 1000 genome, this population is labeled or annotated as Yoruba in Ibadan. In UK Biobank, it's Black African, and Ingnomat is African or African American. So this has led to misclassification and ambiguities related to this population. So the impact is very, let's say, keep the studies not isolated, and we can't do some kind of comparison or mixed analysis. So ontologies. So we had some presentation during the workshop about the ontologies. So the ontologies can be a solution or not for this kind of problem or to fix these challenges. So the ontologies, as definition, are controlled vocabularies. We have a standardized canonical labels and semantic terms. We have a hierarchy relationship between these terms. So I'll give an example as my colleague Elliot presented 2 days ago, 1 day ago, regarding the ontology. So if you pick the finger is part of the hand, and the hand is part of the arm. So this kind of hierarchy definition. So each term should be defined, very well defined, and at the end we have cross-reference. So ontologies, the good thing with ontology that is collaborating with different other ontologies. So we are not reinventing the wheel. We are using terms that already exist and create new ontologies. So why ontologies for population genetics? So there is core benefits and there is scientific impact of developing ontologies. So standardization, so canonical labels, synonyms, and definition for each term. Interoperability, so when we have an ID, a unique identifier, so it doesn't change through the time, and this one can be used machine readable and human readable. We embrace the FAIR principles. You will see FAIR principles during all this Biocuration conference, so findable, accessible, interoperable, and usable, and a semantic clarity regarding the terms that we are using. The scientific impact, so that the reuse and the integration of this ontology and this across different cohorts, clear reporting and reproducibility, enable cross-study and meta-analysis, and reduce biases and facilitate automated data mining and have very impactful publication. So the Africa context, how people came to the African population ontology. So as you may know, the last decade was a very boom for the rise of African population study and genomic studies. So discussion became more insistent regarding the population annotation. So that's why came from the need from the population ontology, identification challenges, and the FAIR principles. So this induced us to use this ontology for the African context. So the ontology offers a formal specification, a controlled vocabulary, and interoperability and reuse. So this you have your structure about how semantic structures our group or member of a population and the genomic data. So you will see the structure of our ontology, so on. Our objective were mainly develop a standardized annotation tool designed to accurately annotate and classify African. I don't like the term classify, so let it annotate population group. We can use this tool for prospective and retrospective studies and embrace the principles, FAIR principles, and cross-study harmonization and metadata. So how did we build AFO? As I mentioned before, so everything started with some kind of community engagement and discussion from Asia Bionet and Asia Africa experts. We did a data collection, which tried to retrieve any data related to African population. We modeled and designed the structure and the hierarchy of the African population ontology. We did some edition, we did a curation, we did an expert review, and finally we released, and we are following different and numerous kind of versioning regarding this AFPO. What type of data we focused on? So we captured data related descriptive languages and genomics. For descriptive data. Mainly we focus on demographic, synonyms, anthropological, and geographic position, languages, names, synonyms, family of language and codes. And finally, genomic data, mainly we focus on mitochondrial DNA and chromosome Y data and haplotypes. So we had two principal classes in our ontology. We have the population class and the country class. So the population class, there is metadata related to geographic association, the position, and the genetic markers. And for the country file, let's say, or our Excel workbook sheet is aggregated genetic information and national demographics, population size, geographic position, and so on. For the quality control, so after collecting and retrieving the data, we are doing cross-verification. So when we have a data, we try to cross-verify our data and our information through different resources. We do a consistency check regarding the terms and the naming. So sometimes you find some inconsistency between resources. We handle missing data by flagging them. We do a different round of curation view, and finally, we had a senior audit or a group of experts that review the data and then validate. Our main data sources, Asia Africa, of course, metadata, scientific literature, public genomic repository, linguistic databases and registry like Ethnologue and 1001 Tribes, and expert input. So I would like to stress here that all our data are public data and all data collected is verified and referenced. So for the technical side, we worked with the platform called Protege Web Protege, which is a tool that allows us to do collaborative editing and collaborative work. So different kind of format and versioning are available using this tool, and we had flexible export, and then we can use online or through a web interface. So for the curation, we had three levels, let's say, or three layers of curation workflow. We do a pre-processing, ingesting data, normalize, and map synonyms. The second layer of curation is editing and logic. So assert classes relationship. This part is mostly technique, And we do a structure control. So we use the ontology development kit for developing and structuring the ontology. And finally, we do the release. We do a peer review and the release and versioning and distribution using GitHub and different other repositories like OboFoundry, BioPortal, and OLS. So the structure of this platform, so the hierarchy, which is the backbone of the ontology, we have the annotation. So each term has some kind of annotation. There is the relationships between the terms is also defined, and finally the description of the structure. This is the representation of the classes. So we have mainly the two classes: geographic location, and we have the list of the country, all the African countries, and you have the African population. So we have the 5 regions, and when you click on one of the region, you have all the list of the population. So we had some, let's say, some challenges regarding this one, because even for the regions, we had problem because there is some kind of ambiguities between, let's say, United Nations and the African Union classification. So for example, Sudan sometimes is classified for the north or sometimes in the center. So the colors is like for the north, there is some population like the Amazigh, the Copt, Yoruba in the west, and Zulu and Xhosa in the south. So the country annotation schema, so the minimum data that should be in the country schema, we have the name, official name, of course. We have the alternative name, we have the codes, and for the genetic information, we look into different resources, and mainly, I think most of our data are related to haplotype for mitochondrial DNA and chromosome Y. So the prevalence, United Nations, AFPO working group, and many, many other resources. For the population, the same thing. We have general information and genetic information. So the general, we have the preferred label, let's say a preferred name, the synonyms, the language spoken, and the geographic position. And for genetic, it's chromosome Y and some variant. So you can see at the bottom of the slide that this parent. So this Yoruba is part of the African population located in Nigeria, Benin, Togo, and we have a cross-ref with the ancestral ontology. So this is so illustrative representation, but you can find another presentations in the different resources. So we have this the definition, the synonyms, location, languages, crossref and provenance. And each data we have the link that take you to the source. Of course, we had a lot of challenges related to this development of this ontology. First of all, we had harmonizing heterogeneous sources. We had a lot of heterogeneous sources and resolve complex synonyms across different dataset and time. We are pretty sure that we didn't cover, I don't, maybe I'm too ambitious. I will say 60% of the population across the continent, but there is a lot of work still have to be done. Mixed population, we had a lot of comment regarding the mixed population because, as you may know, population do not respect boundaries, so we can find the same population in different area and different countries. So we have to deal with this problem. Ethics, yeah. So this is ethics. This is the big, I mean, red flag regarding this when you're working with population. So let me give you some flashback about the story, because as we started, this ontology was labeled ethno-linguistic ontology. And sometimes people didn't like the term ethno-linguistic, so ethnicity, ethno, so it can be stigma, it can be discrimination. So we prefer to have some kind of neutral name, it's population. So we switched to African population ontology. And then the governance, of course, how to deal with data, who's taking the decision, and we had a lot of discussion, negotiation with many people. So I'm going to give you a use case scenario, a hypothetical one. So if you have some, let's say, 5 independent cohorts working on hypertensions, and we say how across West Africa, and they are looking for annotating their population. So they have yeah, when they go to the different resources, they have inconsistency in the labeling. So the solution AFPO is the 3 or the 4 nomination or annotation can be mapped to a unique identifier, which is an ID for the Yoruba population. So this can be then queried through semantic inquiry. So when you go to the ontology, let's say, for example, to OLS, you just put Yoruba, you will have all the information. So we can harmonize the data and do a higher statistical power, fair compliant metadata, and we can cross-study the different participants of the different cohorts. So now we are going to talk to a real-world implementation for the AFPO. So as AFPO is related to the different resources of the AfriGenD, so AFPO is implemented as the African Genomic Medicine Portal, the African Genomic Variation databases also, and the Gen2Fen that my colleague Yasmina presented this morning. So can be also very interesting to see the usage of the AFPO across the different resources. The REDCap, so my colleague Catherine will talk about the toolkit, phenotype toolkit, and I think this is the last presentation. Yeah. So the AFPO list of population is an ID is implemented in the let's say the REDCap phenotype standards and the case report form. So the data is born already interoperable, so we can use it and we can map it and can go back and see the metadata before this ID. We had some, let's say, active engagement with major African national initiatives like Genome Tunisia Project and the Sengen Senegalese Genome Project that we had discussion. We tried to, let's say, start comparing their list of population with ours and see if there is any ambiguities, and we can have the engaged discussion and have a harmonized, standardized list. Yeah, this is some metrics regarding the usage adoption of the AFPO. So I took this graph, let's say the statics from the bioportal resources. So it's around 2,000 since 1 year now, and we have a peak last February, and we have like 473 terms, 700 peak monthly, and 62 properties. So yeah, you can access AFPO on OLS, ABI, Bioportal, OboFoundry, and GitHub. So OboFoundry is one of the main leader of the ontology community. So you can't publish or you can't validate your ontology if you're not filling all the requirement of the OboFoundry community. So our perspective and future direction. So we are having a monthly meeting. Our group of curators are meeting each first Monday of the month. We dispatch the work through the different regions, and we have a report for the data collection. We will try to integrate this AFPO with genomics studies. So I think we already engaged some discussion with the GWAS guys from the EBI. Hopefully we will get some interaction. It can facilitate cross cohort analysis, scientific community adoption. So we try to promote the usage of AFPO for the different scientific community. Yeah, I mean, maybe it's not for soon, but using these tools for the electronic health record into health system will be very interesting too. So I would like to thank the investigator of this project, starting with Asia Africa, Asia Bionet, and now we are maintaining this, let's say, these resources through the AfriGenD project and all the AFPO team. Thank you so much.
Thank you for the presentation, Dr. Melek. Just a quick note, we will be taking or doing a Q&A discussion, panel discussion afterwards. So please hold your answers or questions for then. Up next is Dr. Dominique Anderson from the South African National Bioinformatics Institute, and the talk will be on lessons learned from developing contextual metadata standards for African health data. Thank you so much.
Thanks. Hi, everyone. So metadata standards are usually presented as solutions, right? We have a metadata dictionary, we have controlled vocabularies, we have a schema. But the thing is that we don't talk about the iterative process, which is usually very human and very messy, okay? And this is the development of community-based metadata standards. So over the last 2 years, we've been developing these contextual metadata standards for public health. And I want to talk a little bit about the lessons that we've learned doing this. So first, I need to set the scene. So why are metadata standards for African health data important? African genomics and epidemiological data are globally underrepresented, yet they are contextually distinct. So without fit-for-purpose metadata standards, which encapsulate language, infrastructure, and sociocultural aspects, data cannot be integrated, reproduced, or equitably shared, okay? So we looked at two very different diseases and we had two very different journeys. Neonatal sepsis is a hospital-based disease. It's genomics-driven, and there is a clear unmet need for a metadata standard. Cholera, however, is an outbreak context, very high time constraints, field-based, and it already has entrenched reporting systems. And so we had to look at very different engagement dynamics. Our process for both of these metadata standards were multi-stakeholder working groups, community consultations, and iterative review cycles that led to version 1 of each of the standards. They were shaped not by us, but by the communities who will use them. Lesson 1, language is never just semantics, okay? We had a lot of community debates about over terminology, and these were not distractions, they were the actual work. Community trust, clinical reality, and ontology alignment all come into contact with each other during these debates. I'm going to give you two examples: host versus subject versus patient. Clinical staff might use the term patient, researchers might use the term host, ethics boards might use the term subject. Each term carries different consent, power, and institutional meaning. We cannot pick one without alienating the others. So our solution was to have collection-facing labels that are locally acceptable, mapped to back-end ontology terms which are globally interoperable. Sex versus gender. Sociocultural norms and legal restrictions in some contexts directly constrain what can be captured. And this might even carry a risk for data subjects. A standard that demands ontological, precise gender and sex distinctions may simply not be implementable in certain countries. So standards need to support the contextual flexibility without breaking semantic alignment. So a practical response to this can be layered terminology with explicit synonym mapping, language translation for field materials, and a documented rationale for every term choice. Oop. Why is it not moving? Lesson 2, governance before your schema. Some data fields are not merely technical, they carry political and institutional weight. In the cholera specification, the location field problem exists. District-level location data collected during an active outbreak response carries trade and travel implications. The instinct in our working groups was to immediately remove sensitive fields. The correct answer, however, was tiered access and governance. The field stays because the GTFCC reporting and epidemiological linkage requires it. So what changes is who can see it and under what conditions. The key principle here is to build the access tier frameworks before you build the field lists. Sensitivity is a governance decision, not a schema design decision. Volunteer and community capacity. Standards developed is not just a technical exercise. It is a demand on people's time and on their trust. Planning for realistic participation capacity is as important as planning the data model. Lesson 3. The perfect standard that increases burden will fail. So operational reality versus integration and replacements. So cholera surveillance already has national reporting platforms and WHO-aligned schemas. So introducing parallel data entry risks user fatigue and non-compliance. Adoption depends more on integration than design quality. A good enough standard that slots into existing frameworks will outperform a perfect standard that creates new burden. The field officer test in terms of cholera. Every single field had to answer one question. Can a field officer during an active outbreak without internet or laboratory infrastructure complete this field reliably? If not, it became optional. And this produced a formally approved minimum data set and not an informal workaround. In contrast with neonatal sepsis, hospital-based genomic has very, very good infrastructure. The field officer constraint does not apply. The design principles must be domain-specific, and you must understand your deployment context before making field obligation decisions. Strategies that worked were term mappings and not replacements, minimum information approaches, and explicit interoperability bridges to things like the WHO and GTFCC reporting structures. 4. Standards are social artifacts. Community standards emerge when the value is obvious and immediate, not when it is theoretically important. There is critical asymmetry between the two domains. Neonatal sepsis had a clear unmet need, no competing standards, and the community was more open and motivated. Cholera, there are existing entrenched systems that mean that there was a lower perceived need for something new, requiring a stronger justification for participation. The barriers we encountered were not technical. There were a lack of immediate incentives, completing clinical and outbreak response priorities, and standards fatigue. What worked was anchoring the efforts in clear use cases and empowering internal champions and building on initial existing initiatives rather than starting from scratch. Again, community standards emerge when value is obvious and immediate, not when it is theoretical. So if you are building a standard, your first question should not be, what fields do we need? It should be, who is this for and what problem does this solve? Lesson 5, ontology alignment is bidirectional. Not all clinical meaningful distinctions are ontology ready, and that requires you to adapt your schema and contribute back. Neonatal sepsis classification problem. Early onset sepsis, which happens between day 0 and day 3, versus late onset sepsis, which happens 3 days plus. It's clinically critical. The OBO foundry ontologies encompass entities very well, but can struggle with contextual dependent temporal classifications. In other words, there was no clear term to map to. So we had to do post-coordination, custom extensions, and local control vocabularies. A practical solution could be composite fields, capture the raw data and map to the logic to ensure clinical utility and interoperability. Contributing back. Where existing ontology terms are not sufficient, we need to submit term requests and design patterns. Community standards should feed into ontology development and not just consume it. Lesson 6, the standard is the least of it. The data dictionary is simply not enough. The data dictionary tells you what the fields are, but we received questions from data collectors that the dictionary could not answer by itself. These are not gaps in the schema. They are gaps in implementation guidance. A standard operating procedure, for example, will tell you when to complete them, how to find the right value, and what to do when the value is unavailable. The SOP is the schema. I'm going to be done in a second. The full implementation ecosystem. So beyond the SOP, we have training materials, worked examples, data entry guidelines, validation rules, QC checklists, and curation protocols. Without these, we have inconsistent data capture and quality. The standard exists on paper, but not in practice. So I'd like to call for action to budget time and resources for implementing infrastructure from the start of your project. So the key takeaways: terminology debates are the work, not a distraction. Invest in this from day one. Decide on data governance before you design data fields. Sensitivity is a governance question, not a schema question. Design for the operational context, not the ideal context. For example, a standard which cannot be used in the field will simply not be used. Community buy-in requires demonstrated value. You need to anchor your standard in concrete use cases and find local champions. And start with the SOP, not the data dictionary. Implementation infrastructure is the difference between a standard and a simple document. So I would like to acknowledge these funders and also the working group members and expert stakeholders who helped us develop these. And thank you very, very much for your attention and allowing me to talk.
Thank you so much for the insightful presentation, Dr. Anderson. The next speaker will be Mrs. Catherine Johnston from the University of Cape Town, and she will be talking on the developing clinical phenotype data collection standards for research in Africa. Please join me in welcoming Mrs. Johnston.
Good afternoon, everyone. Oh, I'm very short. Okay, so I'll be talking about what we call the phenotype toolkits. And we have been working on these for a number of years. And so I also really enjoyed the previous talk. I think we have a lot of alignment there. So I just want to present first the harmonization problem. Africa represents 17% of the world's population, 25% of the disease burden. We only have about 3% healthcare workers, and our research, our health output data is small compared to the rest of the world. And the gap in terms of GWAS representation is widening simply because ancestry studies outside of Africa are exponentially increasing. Along with, when we talk about genotype data, we think about African diversity, and the population diversity is critical for identifying genes that help us to find out what does what and what has an impact on a disease, and also for understanding gene function. But when we talk about that gene data, we need to have phenotype data that goes alongside it. If you're a researcher and you are trying to pull together 5 African study data sets, how do you know that those 5 data sets are going to be comparable? If they all say they collect blood pressure and HIV status, and they also collect family history, what are the chances that those variables are going to be able to mean the same thing? The chances are probably low. And so this is what we call the harmonization problem. And we have tried to address this with the phenotype toolkits, which build standards for collecting data to prospectively try and make your data comparable. So when we first started this, it was 2014, and we had the H3 Africa Phenotype Harmonization Working Group. And we started by looking at existing standards to align with. And so we looked at the Phoenix toolkit, and while it's excellent, it was quite Western derived. When we talk about global repositories like dbGaP and EGA and ontologies, many of them don't necessarily manage the transference of data well or represent the diversity that we have. On the right-hand side, you can see Africa is not just one country, it's 54 countries, thousands of languages, diverse populations, and we have a very mixed method of data collection on the continent. And so we were trying to bridge this gap by firstly using and adopting existing standards, adapting them where we needed to, and then also developing new ones. So what did we build? We did 11 toolkits and 82 data collection protocols. When I say data collection protocols, you can consider this to be like case report forms that projects, genomic research projects, could go and apply to their data collection. We adopted 38 existing protocols from Phoenix as they were. We also adapted 17. An example of this would be family history, where terminology about nuclear family, for example, in the Western world, it does not necessarily align with the context that we're in. And then we also created 27 novel protocols where we couldn't find an existing one. And the process that we used was first to inventory existing ones, to figure out whether we needed to use something, adapt it, or create a new one. And then we created the tool itself and integrated it and developed it in REDCap. And these were sent out to field experts, both internally with the H3 Africa Consortium and experts outside of it, to confirm that what we were collecting was the essential variables. And then we mapped those variables in all the data dictionaries to existing ontologies, and also created guidelines to go alongside the toolkits for those collecting the data, so that we weren't just providing a tool to collect, we were also providing the methodology behind the data collection and an explanation for users. So we have 11 toolkits. We start with the core phenotype, which addressed adult and pediatric data collection. And then we also have 10 other domain-specific toolkits. Each of them has everything you need to swiftly spin up a REDCap project. So this means it includes a REDCap XML file, the data dictionary, guidelines, and I think that's it. And then so all the variables are also compatible, so you can pick and choose what you need. And although we don't recommend it, you can remove or add things, but we try to maintain that what we've put there is what we consider essential for genomic research. We also have a microbiome and COVID-19 toolkit that are standalone. So this development continued under H3O Bionet, and when the African Genomics Research Hub came along and H3O Bionet had retired, we picked this up to continue development with many of the original members who were in the first set of development. And to really make these standards truly inclusive and to consider the diversity on the continent, we decided that a key thing to do would be to translate them. So we benchmarked some AI tools, because now AI had come along and that made this a doable thing, and something that we could do quite quickly. So we settled on Google Translate after trying various tools, because we wanted a tool that worked well with different African languages. And we mainly settled on that because it was free and it did the job, and it also worked in the spreadsheet environment that REDCap data dictionaries came out. So we'd made those translations using AI. We then handed them over to members on our team who were native French speakers to validate. We applied changes we needed to, and then thankfully in 2025, REDCap had made some major advances in its translation multi-language management module. And so that also came along at the same time. They produced a great guideline for how you could integrate this in REDCap. And so we did that and released the toolkit in our GitHub repo on AfrigenD. I think it's important to recognize that it requires domain expertise to understand the terminology sometimes. And it was really important that it was a community effort. All right, so what did we learn? Global biocuration isn't just about collecting the diverse data. You also have to have the standards that allow that to take place. You need to make sure that you're not excluding excellent research because you don't necessarily have standards that work in different linguistic or cultural contexts. So we can do this by broadening our participation in curation activities to address the gaps. We can make sure that the tools that we build are robust enough to work in different contexts. And we also need to remember to strike a good balance between the goal of standardization and harmonization without losing the features that we need to still collect to maintain that context. So, African data collection and digitization, we know, is far behind. It's quite fragmented. People have different methods for collecting data on the continent. But with AI's advancements, we can now almost leapfrog over systems that we don't need to put in place that maybe didn't address the needs in Africa. And we can now jump straight into designing systems that integrate AI and align with global standards whilst we continue to develop ones that work in Africa. Data needs to be collected in ways that makes it interoperable and fair, and so it needs to be curated from the onset wherever possible. And I'd like to extend an invitation to the biocuration community. One of the most important things is that you have that community involvement. And so we would really invite for feedback and updates and logging of issues on our GitHub repo for those who use the toolkits so that we continue to build standards that work for everyone. Thank you.
Thank you so much, Mrs. Catherine, for the wonderful presentation. Last but not least is Dr. Eddie Lulamba, and he's from the South African National Bioinformatics Institute, and his talk will be on standardizing contextual data specification for malaria molecular surveillance for an African perspective. Please join me in welcoming Dr. Eddie.
Hello, everyone. My name is Eddie Lulamba from Zambia. Today, I'll be speaking about the development of the malaria metadata standard, more about the development process and what it is. So the topic is standardizing contextual data specification for malaria molecular surveillance, an African perspective. We all know that malaria remains one of the most significant public health challenges globally, and especially in Africa. And then malaria molecular surveillance enables early detection of resistance markers and better understanding of transmission. But this requires robust interoperable data system across public health system and research to be able to collect data, helping to answer genomic surveillance and epidemiological studies questions, things like who was infected, when and where the infection occurs, and what are the factors contributing to the infection. And currently, there is no single widely adopted standardized metadata framework that integrate epidemiological, clinical, geographic, laboratory, and sequencing metadata. So there is a need of a standardized contextual data specification for malaria molecular surveillance that harmonizes these domains while supporting interoperability, reuse across public health systems and studies. However, to be able to address this in the global south in Africa, there are persistent barriers preventing to actually do that. And these include things related to data sharing and standardization. So among those barriers, we have challenges related to data ownership, attribution, and trust within open science frameworks. Things like concern about data sovereignty, especially when the data is to be sent off continent. So people might be concerned about onward use of those data, about losing access to own data or just being scoped. And things like informed consent might not be sufficient, the sensitivity of genomic data and associated metadata, and mechanism for cost recovery is not. There's no mechanism for cost recovery, and the lack of benefit sharing is not even inculcated. So the other concerns that are limiting the us to do that is limited adoption of data standardization practices, especially regarding the malaria. So this is because of diverse data sharing needs. So in Africa, malaria data is often collected to be shared locally with neighboring institutions rather than globally. So with little implementation of interoperable practices. Secondly, the high malaria burden within limited resources. Most pathogen data standard metadata standard that are being developed are from high income countries, which may not be practical in the African context. When we look at the number of metadata fields, maybe also the level of technical complexity that may not be applicable in certain low settings environment in Africa. And as a result, in many settings, limited to no data is being collected with the purpose of global sharing or reuse. So how do we move from collecting almost nothing to collect something? So to address that, we developed the malaria contextual data specification called Malaria Community Metadata Standard. And the development process involved, we followed established best practices for pathogen genomic data standardization and emphasized reuse of existing ontologies and metadata framework wherever possible. And these frameworks are ISO aligned, ontology informed, developed using open source semantic standards to support machine readable, interoperable contextual data capture. So how did we establish that standard? So it was established through scientific evidence review and structured community consensus under the Pathogen Genomic Data Standardization Initiative in Africa, which is a collaboration between Africa CDC, phage, public health laboratories, and the GenAP network. And this was done to actually promote inclusive participation, regional leadership, and trust-based approach to data governance and sharing. So this is just what I've explained. So the process started with reviewing existing literature and standard, and then compiling and mapping malaria data collection practices against the widely pathogen standard, and then curate them through collaboration with our different partners. We're looking at things like scientific accuracy, interoperability, and field feasibility. And afterwards, we came up with the standard. So what it is, it's a framework that consists of thematically grouped modules comprising standardized metadata fields and control vocabularies structured into two complementary subsets, one addressing human clinical and one health samples, and the other one on mosquito vectors and topological data. And the package includes an Excel data collection template, a field reference guide, term reference guide, and some curation guidance to support consistent implementation and data harmonization. So these are some of the evidence that we use to develop the standard. So the data sources, those are the data sources that we used, and mapping the malaria data collection practices to the widely accepted. Here's my pointer. So these are the malaria data collection practices that we compiled, and then we mapped them against some of the widely used standards, including the phage standards for SARS-CoV-2 and Mpox, and then some of the repository requirement from INSDCs. And then after doing that, we came up with so we compile all of these and then do the curation, and then came up with over 210 metadata fields, which were grouped into 14 categories or modules. And those modules collected information regarding specific information. So like the first one was collecting the database identifiers, and the module 2, for instance, was collecting the sample collection and processing information, and so on. And this is the figure showing how we mapped the malaria standard to the to the widely used standard. As you can see, in green, we have the malaria human and one-off samples. And here we have the malaria mosquito vector samples. And you can see how the field, the modules matches the most, the widely used, like the SARS-CoV and the wastewater and the mpox. So all these fields were enriched or reduced to adapt to reflect the malaria genomic surveillance and AP studies need, the data need. So this is how it looks. So this is the field reference guide. The first column is to collect the contain the metadata field, the metadata field name, which are grouped into modules. So this one is the sample collection and processing modules. To each metadata field, there is an ontology ID, which is linked. And then we have determined what type of variables and the definition, the guidance, and the examples. So in conclusion, so we're hoping that the malaria community metadata standards will provide a foundation for harmonized malaria molecular data sharing and surveillance across Africa. And because it is driven by regional collaboration, it will enable data interoperability, strengthen governance, and enhance public health response. I'd like to acknowledge my institution, all the founders, and all the collaborators involved in the development process, and for this conference organizers for this wonderful opportunity. And thank you for your attention.
Thank you for the presentation, Dr. Eddie. Now I ask that all the speakers join us so that we can open up the stage for questions from the audience. Can we have a mic over here? Yes.
Right. Okay. So there was a lot of talk about data harmonization there, and this all seemed like good old-fashioned, let's write definitions and really try and work it all out. But I suspect in the coming years, it's going to be a lot of AI-driven harmonization, and this is kind of for anyone, what your thoughts are on, is this going to be sort of a miracle or a total disaster? Yeah, what do you think?
Thanks, Alex. That's also why we work on the harmonization tool that Atwine presented. So I definitely think that AI is going to hopefully help with the harmonization of data. I think that the curation and making sure that the data is what it says it is still an important part. We're not going to be able to feed it into AI if we aren't sure about what it is. So I think, I mean, the hope is that AI is going to enhance this process and speed it up. If you try and do data harmonization manually, it is a miserable experience. So my hope is that the development that's going on in all these different quarters, both with building ways to curate the data better, and also building tools for how we can get AI to process it well is going to solve that massive problem.
Yeah, let me, and then, so in my experience, so Catherine presented the translation on French. So I've been involved on previous experience with ontology to translate an ontology for the sickle cell disease ontology. So it's way before the AI. So it took us, yeah, I mean, more than 6 months to do it. I mean, like hundreds of terms that should be verified, translated. Yeah, we tried Google Translate. We do it by ourselves. Took us 6 months. How many days did you take, Bobby, to translate this? 2 minutes?
Yeah.
Yeah. I mean, we did just after that, we did some kind of verification. It took us, yeah, maybe a couple of days. So we can see, as you presented in your talk, so there is some boom into, maybe it's better, for sure it's better, but we need some kind of, yeah, big vision about it, how to deal with it, not get into it and dig to it, we can lose a lot, so yeah.
Just based on that as well, I mean, I think when we developed this, the metadata standards, is we're using community to do this, right? And the thing is that like AI doesn't have an understanding of, like, it's not sitting in the room with your community and having these debates about, oh, I don't like this term, or, you know, what does this mean? Where is the ambiguity? Because the thing is, we're linking these things to ontologies and things like that. So I think that AI does have a place, but it's going to augment that, and it's going to perhaps like increase the time that we can find synonyms for these things. And, you know, so taking away that very, very manual process. But the reality is that you still need to sit with the community, and the community is not AI. So there is place for AI, but there's still that sort of manual section that does have to happen.
Yes, sorry, just a second. There was a question at the front. Would you mind just introducing yourself briefly?
Thank you very much for all of you. Thank you, Malik, for your insightful presentation. My question, it's a comment more than question. As part of AFPO sub-project, we see integrating linguistic and historical expertise as a possible way to improve population labels in African genomics data.
Yeah, so we try to reach out to anthropologists. We try to reach out to expert in history. So yeah, I mean, most of the validation should be at the end visioned by experts. So we do not have a lot of contacts with them, but as we mentioned, my colleague mentioned regarding the community engagement, I think being in touch with experts, being in touch with representatives of some communities and population would be very interesting to improve the quality of the FPO. For the moment, we are just reporting data that exists, but we are open to, for more layer of information and data from the experts. Thank you, Laura.
Thank you.
Maybe just to add on that, to the standard we're developing are supporting the multilingual translation, because we're using what we call the IDs. So people can actually use whatever term they want locally, but keep the ontology. Okay. So when integrating the data, we can still know exactly what column to pick and to make. So we, integrating that in the standard. Yeah.
Thanks. Oh, yeah, go ahead.
It's really a quick comment on the first question. I think Dominique's talk didn't mention AI once, but it should have been subtitled why AI might not work in data harmonization.
We have a question at the back.
Hi, yeah, Dr. Dominique Anderson, lovely presentation. I was looking at lesson number 5, SOPs first, not data dictionaries. As somebody that's been obsessing over data dictionary viewers and how we can better communicate our data models, it might have been something that I've overlooked in designing some of our user interfaces. What concrete examples do you have for some of those SOPs that need to be communicated alongside the dictionary?
So in our case, like when we're looking at these SOPs, we're looking at an overall thing. So first of all, an SOP can't be 400 pages long. Nobody's going to read it, right? But we're working with public health. So public health is actually like attuned to using an SOP to guide their work, right? So that's the one thing, is we're not introducing SOPs where people are completely unfamiliar with SOPs. And what we actually do within our working groups is we also allow our community to help develop the SOPs as well, right? So we're not just sort of going, oh, we'll do this and you've got to read it and you've got to sort of like, you've got to follow it. And I think the SOPs are quite important because the fact that like we had things where we would do some beta testing, we'd send out the data dictionaries and the Excel collection templates, and we'd have people coming back to us saying, okay, but like, I don't know what's put in this field. What happens if like, I don't have this information? These kinds of things. And you can like really, really structure these into SOPs, and you can define your SOPs as to what aspects you want things to answer. So your community is going to come to you and say, oh, I didn't know what happened here, or these are concerns that I have that I couldn't find in the data dictionary, or this thing is ambiguous and I didn't understand. And you can actually structure your SOPs so that you're addressing each one of these individually. And I think that that's the way to really sort of to take it on, is to say that like this SOP is going to address this question, this SOP is going to address this question, and this SOP is going to address this question. Does that answer your question?
It does, and I will follow up with you either today or tomorrow to further the conversation. Thank you.
Do we have any more questions?
I have a comment.
Go ahead.
Yeah, that's quite interesting. We saw two presentation or talks regarding developing standardized phenotype metadata. So we have Catherine who's working on human, and our colleague who working on pathogen. So yeah, you should. This conference can be a good opportunity to collaborate and make something related to human and pathogen, as you mentioned, One Health. So when we talk about One Health, we'll talk about the circle between the human pathogens and environment. So yeah.
Do we have any more questions or comments? Maybe from the speakers? You're fine? Okay, then I think it's appropriate to close down this session and the day. Thank you so much to the speakers for this insightful session. I just ask that you stay behind for a photo, please. And then can I